So it's my privilege to welcome Dr. Ramchandra, Director of Kidwai Institute of Oncology, who has gracefully consented to deliver the inaugural address despite his hectic schedule. And also I welcome Dr. Hanumanthachar Joshi, Principal of Sarada Vilas Pharmacy College, who will be delivering the ongoing opening remarks, and Dr. T.M. Pramod Kumar, Principal of JSS Pharmacy College, who will also address this virtual gathering of enthusiastic and eager academics. I also welcome all the resource persons who will be sharing their knowledge and expertise with us today. Mr. Rishi Kumar, from the National Coordination Center, the Pharmacovigilance Program of India, Ghaziabad. Dr. A. Pramod Kumar from the CH Institute of Pharmaceutical Sciences, Guntur. Dr. M. Ramesh, Dr. Sriharsha Chalasani, and Dr. Juni Sebastian from the JSS Pharmacy College, Mysuru. Dr. Priyank Tripathi from the Healthcare Global Enterprise Limited, Bengaluru. And Dr. Roha Dholakia from the Kidwai Institute of Oncology. Lastly, and most importantly, I welcome all the delegates who have registered to this online workshop in such large numbers. Thank you.
Thank you so much, sir. We also welcome you for this workshop on pharmacovigilance. Now I call upon Dr. Hanumanthachar Joshi, Principal, Sarada Vilas College of Pharmacy, Mysuru, for the opening remarks.
Good morning, everyone. Respected dignitaries here, we have assembled for the inaugural session. A well-known doctor and more than anything, a very wonderful human being, Dr. Ramchandra sir, and our very good friend and dynamic principal of JSS College of Pharmacy, Dr. Pramod Kumar, and an young person and who has making very good marks in the field of pharmacovigilance, Mr. Rishi Kumar, and our own friend Dr. Dattatri sir, and my friend Dr. Rosie. It's really a pleasure to be a part of the inaugural session of the pharmacovigilance workshop, which has indeed very much required for the young and budding people who want to enter this profession. The burden of adverse drug reactions in the global scenario is high, and accounts for considerable mortality and extra cost to the patients. In developed countries, 4.9% of the total hospital admissions were due to the ADRs. If you look at the entire the population, around almost 5% of the admissions are because of the adverse drug reactions. This is the data which have been received. And ADRs are very common in the healthcare system. And in the United States of America, ADRs contribute to 3.4 to 7% of the hospital admissions. And in UK is 1.9%. And in certain countries, it is almost 10%, which is really a cause for concern. And in India, with a current population of more than 1.3 billion, and which is the fourth largest producer of pharmaceuticals in the world, with more than 6,400 licensed manufacturers and over 60,000 branded formulations in the market. Therefore, medicine safety monitoring is an essential part of the healthcare system and for high quality medication. Since safety monitoring of medicines as an integral part of the clinical practice, the Ministry of the Health and Family Welfare, Government of India, has launched a program called as PVPI, that is Pharmacovigilance Program of India, on which I think Mr. Rishi Kumar will be explaining more when he'll be delivering his talk. And the main purpose of this pharmacovigilance program is to inspire the confidence and trust among the people, because very well there are so many ADRs which are being unreported and which are there, which is a common part in the practice. And Indian Pharmacopoeia Commission under MHW has been functioning as the National Coordination Center, which Mr. Rishi Kumar represents. The major objective of this workshop is to brief and make the delegates understand the importance of pharmacovigilance and to encourage ADR reporting in their setup. That's what the expectation is. And we are very happy to associate with Kidwai Memorial Institute of Oncology in hosting this meaningful e-workshop. Kidwai Institute of Oncology is doing an excellent work under the dynamic leadership of Dr. Ramchandra. I was just talking to him yesterday for a very brief time, and I understand that they have already planned and established a state-of-the-art drug testing laboratory, which will be helpful for all the people. And I sincerely appreciate Dr. Avinash, the person behind the show, the senior medical officer, KMIO, for his dynamism and sincere efforts. And the support of Mr. Rishi Kumar from PVPI Ghaziabad is really appreciable. In a short notice, he accepted that. And there are now no words to appreciate Dr. Pramod Kumar and his team. Very active and dynamic team for part of this very workshop. And our one more friend will be joining, Dr. Pramod from CHIPS and Dr. Tripathi from HCG. And I am very sure that the deliberation of today's workshop will be beneficial to the delegates, and hopefully Let us meet soon physically rather than only online. So with this, I am really happy to be a part of the inaugural function, and thank you so much.
Thank you, sir. Thank you so much. Now I request our guest, today's chief guest, Dr. C. Ramchandra, Director, Kidwai Memorial Institute of Oncology, Bengaluru, to deliver the inaugural address. Over to you, sir.
Very good morning to everyone. At the outset, I wish a good health to everyone in this pandemic, COVID pandemic. Surviving is a big issue. And I thank Dr. Avinash is behind this program. In fact, he is instrumental in me bringing to this, this forum. And he was very much concerned about the pharmacovigilance program. And he is actually the man behind pharmacovigilance program in Kidwai. We have about 3 pharmacies, and we have a patient load of about approximately 1,500 patients every day. We see about 20,000 new cases every year and more than 4.5 lakh follow-up cases every year. So we cater patients from Kanyakumari to Kashmir. So there is no differentiation here, and no patient goes back without treatment, whether they have money or no money. See, money is a secondary issue here. Humanity is the most important issue here, and we treat everyone with equal, importance. With this, COVID pandemic, most important thing in pharmacovigilance is in developing vaccine. Everybody is talking about the development of vaccine. It is not easy here. Many times, this has to go through pharmacovigilance program also. And I understand that, people talk very high about vaccine. Unfortunately, the issue is here whether they have a long-lasting immunity with the COVID vaccine. I myself, being a patient, just come out of the ICU nearly about 6 to 8 weeks back. I tell you, it is a terrible experience. I have never experienced in this issue, and being with a man with no comorbidities, I struggle like anything for nearly 2 weeks. So ladies and gentlemen, I thank Mr. Dr. Hanumantrao, the principal of the Sarada Vilas Pharmacology College, Dr. T.M. Pramod Kumar, principal of JSS Pharmacology College, Dr. Nadav, the president of Karnataka Government Medical Welfare Association, Medical Education Welfare Association, Mr. Rishi Kumar, the Scientific Assistant of Pharmacovigilance Program of India. And at the outset, the most important thing is I thank Dr. Avinash for being, initiative in this program. And I welcome all the faculty and the delegates on this. I wish everyone benefits out of this discussion and the rich knowledge of all the experienced people. Ladies and gentlemen, many developed countries have their pharmacovigilance programs following the thalidomide incidents in the scandal in 1960s. The whole issue, thalidomide was issued for many reasons, better, and we still use thalidomide today. One of the reasons, I spoke about thalidomide is re-established, and we are using because we found out the serious adverse reactions and the usefulness of the drug. The most important thing I advise every pharmacist and every doctor in this country is the ethics and principles. Please don't leave ethics and principles. You always treat anybody, whether it is an outsider or an insider or somebody else unknown or known, they are like my family. You should always think every religion is equal in this country. Every person is equal in this country. Please treat everyone with equal importance and, balance everything. Most important thing there are a lot of students I have registered. Please, most important thing is ethics and principles, especially in pharmacology where there is a lot of money is involved. Don't go behind money. Money is a secondary issue here. Forget about this. Follow the ethics and principles. Don't ever leave ethics and principles. Ladies and gentlemen, the Central Drug Standard Organization established the present pharmacovigilance program in India in 2010, and the program was integrated with the government legislation, a regulator as a leader, and a research center as part of Indian Pharmacopoeia Commission. In 2018, there were about 250 centers in India is capable of responding reports of serious adverse reaction. The most important thing is I, being a principal investigator for many clinical trials, lot of the foreign companies, the multinational companies come to India because we have a rich resource of the patient load. These patients, many times, what we think is many times, the second line, third line and fourth line drugs are not free and they are very expensive. We have an association, our institute is being completely adopted by the Ayushman Bharat and Karnataka Arogya Shri scheme. We call it as Vajpayee Arogya Shri scheme. In this, we give only first line and part of the second line treatment, whereas third line, fourth line, and sometimes fifth line or sometimes till death. patient needs treatment. How will they get this treatment? The advantage of clinical trials is this. Many times people think that, doctors actually doing clinical trials go to abroad, they travel to abroad and they make money. This is a secondary issue according to me. The most important issue is the benefit to the patient here. These drugs are actually, cost in lakhs many times, and the company sponsor free drugs. Not only free drugs, they also give the travel allowances, the investigation allowances. Everything is free of cost. This is a major benefit. So the foreign companies and the most of these the MNC companies come to India because we report serious adverse reactions, and it is a safety haven for them to conduct trials. This should be facilitated. More and more clinical trials to be conducted. Unfortunately, because of some issue, the clinical trials were closed for last 3 years. But now I'm reopening clinical trials for all the multinational companies across the world. Why pharmacovigilance is very important? I think you all know about the carbamazepine drug, which is being used for trigeminal neuralgia, where you get a throbbing headache. This drug is being used in the European countries and many multinational companies. Unfortunately, being Indians, including me, we have a habit of extrapolating Western literature to India, but it doesn't work. Our scenario, our genetics is totally different from the Europe and USA genetics. And unfortunately, they found out because the pharmacovigilance company, we could find out carbamazepine can cause serious adverse reactions in these companies. We did identify in this, but still selected cases we do use. So these are all the advantages of pharmacovigilance company I'm trying to talk about is. The establishment of pharmacovigilance has made the more attractive international companies for clinical trials, and I wish every center should have a pharmacovigilance company. It is not only every hospital, every clinic should follow the pharmacovigilance effects. The problem with the India is there are many companies, many clinics, many hospitals where it has not been reporting serious adverse reactions. For example, I'm not against Ayurveda. I, being a true Indian, India has a rich resource of Ayurveda. Unfortunately, Ayurveda, there is no chemical structure, there is no level 1 evidence, and no clinical trials are being conducted. And you cannot do a randomized study without a chemical structure. I think you know better as pharmacists, better than any doctors. The basic reason, we had an Ayurveda unit. I had to close down the Ayurveda unit because they were mixing with allopathy, and that many people have complained serious adverse reactions, including the mortality in this issue. So I had to close down because of this issue. Why I'm saying because just because we have a rich resource doesn't mean that we utilize free of cost. The most important thing is safety of the patient and the effectiveness of the patient. This needs a series of trials starting from phase 1 onwards. And our drugs, every time we conduct a drug, it has to go through a clinical trial, and it has to go through phase-wise manners, and it has to establish itself in this country. So I don't think that I'm against Ayurveda, and I, as a person, as a surgical oncologist, I being a surgeon, so I've been practicing for the last 27 years in this hospital. I must have done some thousands and thousands of surgeries, including open laparoscopy and robotic surgeries, and we have everything here. And the lot of politicians and senior bureaucrats also were angry with me because I had closed down the Ayurveda unit. Unfortunately, I'm not against Ayurveda. I am for it, but I told them to establish a center of Ayurveda outside Kidwai hospital. You make a 100-bed hospital wherever these drugs, our allopathic drugs exhausted, whether it is first line, second line, third line, fourth line. When there's a terminal ill patient, when you cannot do anything, such patient will refer to you because there is no other resort for them. Otherwise, you cannot use Ayurveda as a first line drug, basically because of the severe adverse reaction and no clinical trials being done and no level 1 evidence for this drug. The program also, the clinical, the pharmacovigilance program, it coordinates with the WHO in every corner and every step in this issue. Places like United States and Europe have a pharmacovigilance system which are developed well, but in Indian scenario, it is still at the infantile stage in many places, especially in rural areas, and it is well established in the urban areas. Our pharmacovigilance company, the state, program in Kidwai Memorial Institute of Oncology is very strong, and we are happy to be honored. It's a really honor to host this program, and I'm really indebted to all the persons involved in this issue. And I thank again Avinash. He is instrumental, and he is the man behind this. And for about last few months, he said, "Sir, we have to conduct this." He said, and henceforth, Kidwai will coordinate with you in every step. As long as we are here, we'll support you in every endeavor and every respect. Most important thing, according to me, patient is God. So the safety of these drugs is very, very important in this issue. We have actually, I thank the Sarada Vilas College and JSS College and the Pharmacovigilance Program of India for coordinating with Kidwai in this regard, and the Karnataka Government Medical Faculty Association in this issue. We are happy that the resource persons from the National Coordination Center, the Pharmacovigilance Program of India, Central Drug Standard Control Organization, New Delhi, and the Regional Training Center of South Zone, Pharmacovigilance Program of India, JSS Medical College Hospital, Mysuru, in association with, of course, again, I tell you, Sarada Vilas, the College of Pharmacy. I expect it will enrich the knowledge of the experienced people, and they'll make them more aware of the safety of the drug, ultimately of the safety of the patients here. And I'm confident that the deliberation of the workshop will be useful to the young doctors, young pharmacologists, and all the people involved in this issue, all the delegates, I mean. So the people who are involved in researchers, the health professionals, and benefit in setting up a new pharmacovigilance center in their group. I wish you best of luck. Have a nice program. Thank you.
Thank you, sir. Thank you for your kind words and well wishes for this workshop. Now I request our guest of honor, Dr. Pramod Kumar, Principal, JSS College of Pharmacy, JSS Academy of Higher Education and Research, Mysuru, to speak few words about this orientation workshop on pharmacovigilance. Over to you, sir.
Thank you, Dr. Rosie. First of all, good morning, everyone. Am I audible? Yeah. I bring here the greetings from our Pro-Chancellor, Vice-Chancellor, and Register of JSS Academy of Higher Education and Research, and also the blessings of His Holiness Swamiji for this very meaningful workshop. At the outset, I think it is a wonderful workshop that Kidwai Institute of Oncology has organized, and a big thank you for Dr. Avinash and Dr. Ramachandra, the director, who are the men behind all this conduction. Thank you very much, and it is a very meaningful one. We have a good facility over here, and you have identified a very resource persons from different parts of the country, and we being from the JSS, most of the speakers are from our own colleagues, and I'm very happy that it is we are also a part of this workshop. And to just give you an overview, the Department of Pharmacy Practice, when we started in 1995, we were the first to report the adverse drug reaction to Uppsala Monitoring Center at WHO, and that was a time when even PVPI, Pharmacovigilance Program of India, was not constituted. And that was a credit to what we have, and even till today we have the highest number of adverse drug reporting, and now the doctors have widely appreciated the services that are extended by our pharmacists in not only in JSS Medical College and Hospital, even we have extended our services to the neighboring hospitals like Bharat Cancer Hospital, Columbia Asia, then Asha Kirana Hospital. So like that we have expanded so that the services should be available to all the different hospitals as well, not limiting to only our hospital. And for all the good services that our team was doing, they have got a recognition from PVPI Center that it is a regional training center of pharmacovigilance in the South Zone. And again, we are very happy and take the pride that we have trained more than 1,000 doctors, pharmacists, and healthcare professionals, and conducted number of workshops and seminars across the South India. And we also conducted a number of research projects pertaining to the pharmacovigilance at the level of masters, at the level of PhD, and even the doctors have contributed in a very big way And I'm very happy to see here that our pharmacy colleagues and staff and students are right now taking part in the phase 3 and phase 2 and phase 3 clinical trial of Covishield vaccine, which is right now a buzzword and where the whole of the globe is looking at. And we are very privileged for being a part of this clinical trial thing. And even I am a volunteer as well. And there cannot be other person than Dr. Ramchandra, who has undergone the real pain on the suffering of this COVID, and you have come out. I pray for God that you have a very good health and serve the country and serve the patient, because according to you, patients are God, and certainly you have inspired young minds with all the ethical principles, which is very much missing many times in the present day. And I think as Dr. Hanmantacharya Joshi said, you are a true human being, and also love for the country that you have, which will certainly lead to Atmanirbhar Bharat, which our Honorable Prime Minister is always talking about. And this workshop would be very meaningful. And from the College of Pharmacy, Mysuru, and the Department of Pharmacy Practice, if Dr. Avinash and any other team wants any type of support, let us collaborate. Let us see how we can take it forward and come out with a wonderful publication, research aspects. Let us forget, even if there are any small elements of differences among the doctors, pharmacists, nurses, or any other healthcare professionals, and even then we can move forward. And for all these things, we have a wonderful support from the Pharmacovigilance Program of India. They have been highly supportive for all of our initiatives. And thank you very much. I wish that this workshop would be very useful. And we look forward to have more and more interaction with Kidwai Institute and Dr. Avinash in particular. Anytime, you are most welcome, Doctor, and we are there with you. Thank you. Wonderful. Thank you very much. And wonderful that I was given a chance to be a part of this inauguration. Thank you very much, sir.
Thank you so much, sir. Thank you for encouraging words. And I thank Dr. Ramachandra, Dr. T.M. Pramod, Dr. Dattatri, Dr. Hanmantacharya Joshi for sparing your valuable time and blessing this event. Now, without further ado, we will turn the time over to today's first speaker, Mr. Rishi Kumar, Scientific Assistant, National Coordination Center, Pharmacovigilance Program of India, Indian Pharmacopoeia Commission, Ghaziabad. I take this privilege to introduce our first speaker, Mr. Rishi Kumar. Rishi Kumar is a pharmacist by profession and has more than 14 years of experience in academics, research, and pharmacovigilance. He is an alumni of BIT Mysuru, Ranchi. Presently, he is working as a scientific assistant in Indian Pharmacopoeia Commission, Ghaziabad. He has published more than 15 papers in various national and international journals of repute. He has been a part of training teams since last 7 years and trained more than 450 people in the QMS pharmacovigilance. He has also been a part of workshop cum training program conducted by World Health Organization in collaboration with Pharmacovigilance Program of India for healthcare professionals as well as medical professionals involved in revised National Tuberculosis Control Program, National AIDS Control Program, and National Vector-Borne Diseases Control Program. His interest is to be a part of effective and robust pharmacovigilance system and to provide better inputs in existing healthcare system. Now I request Mr. Rishi Kumar to deliver the talk. Over to you, sir.
Thank you, Dr. Rosie, for introduction. First of all, I would like to thank Dr. M. Ramesh for giving me opportunity to be part of this program. And I also thank you. Thanks the organizers for inviting me to this platform. Also, I also thanks to Dr. Dattatri A.N., Dr. Hanmantar Joshi, Dr. C. Ramachandran, Dr. T.M. Pramod Kumar for their valuable words in the inaugural session. So with this note, I start my presentation on the topic recent updates on pharmacovigilance program of India perspective. So I'll start my presentation. The content of my presentation will be mandate of Indian Pharmacopoeia Commission, need of pharmacovigilance and what is pharmacovigilance. Next, Pharmacovigilance Program of India, its establishment, how it has been established and how it has been transferred to Indian Pharmacopoeia Commission. Next, I'll take a look on the organogram of Pharmacovigilance Program of India. Next, we'll go to the functioning of Pharmacovigilance Program of India, and the last, we'll discuss about the outcomes of Pharmacovigilance Program of India in the last 10 years. So, before going into the details of pharmacovigilance, I would like to give some positive inputs regarding the COVID-19 pandemic, as we know that we are going through very hard phase due to this COVID-19. And it is a positive news. I just got it from the Press Information Bureau of India that cases per million in India is lowest in the world, as well as the death of the people occurring due to the COVID-19 is also very less. It is 87 per million in India. If you want to read this article, it is available on the website of the Press Information Bureau of India, and you can go through this. So, coming back to our topic, that is matters of Indian Pharmacopoeia Commission. Basically, Indian Pharmacopoeia Commission is an autonomous organization. It is working under the aegis of Ministry of Health and Family Welfare, Government of India, and it is having 4 basic mandates. First is the Indian Pharmacopoeia, publishing of Indian Pharmacopoeia. Indian Pharmacopoeia is a book of reference, book of standards, which is being used for quality control and quality assurance of medicine in pharmaceutical industries. Next, Indian Pharmacopoeia Commission provides reference substances. Reference substances are those substances which are used as a reference for comparing the manufactured drug in pharmaceutical companies. Third, we are responsible for release of National Formulary of India. It is a book of reference for medical professionals and other healthcare professionals for rational use of medicines. The 4th mandate of Commission is Pharmacovigilance Program of India, and it has been if we talk about the definition, as we all know that it is we can remember this definition with 4 letters, that is DAUP. This pharmacovigilance is the science and activities related to detection, assessment, understanding, and prevention of adverse effects of medicines or any other medicine-related problem. So, why we need pharmacovigilance? It was very beautifully highlighted by Dr. Hemanta Joshi sir regarding the percentage, how much economic burden is putting on the government of various countries and patients. So, to protect the patient from unnecessary harm and increasing healthcare costs from medicines, we need pharmacovigilance. Adverse drug reactions are a huge burden, as Dr. Hemanta Joshi has given some references of data that it is giving. It is responsible for maximum. It is responsible for a significant number of admissions in hospitals. Insufficient evidence of safety of the drug from clinical trials. It is for that we also need some pharmacovigilance, and as in our last mention that during phase 4 we need to continue with the pharmacovigilance. Early detection of new adverse drug reactions on patient subgroup of exceptional sensitivity, say for example pediatrics, geriatrics, pregnant women, because during clinical trials these group of participants are very less in number or significantly no, because it is ethically it is not good to involve pediatrics and geriatrics and pregnant women in clinical trials. So step by step, this study is being done later on after approval of drug when we are doing the post-marketing surveillance. In that case, it has become very important for the healthcare professionals to do the pharmacovigilance studies on pediatrics, geriatric and pregnant women and be vigilant while we using the drug on this special group of population. Next is promoting rational use of medicine and medication adherence. It is also required for that. The pharmacovigilance is also required to support the regulatory agencies in decision-making process for safe use of medicine. As one of our eminent speaker, Dr. Dattari A.N. also said that because whenever we are looking for any regulatory decisions, we are looking to our best. But at present, we can say that India is having pharmacovigilance program of India, and we are having more than 4 lakh of adverse drug reactions report in our database, and we can analyze any drugs through based on our database, and we are not required to look toward West for taking regulatory decision, because now our structure of pharmacovigilance is that much good that we can do our studies, we can do our science evidence-based study, and we can take decisions. Next, boosting public confidence. It is very much required for boosting public confidence in the medicine, safe use of medicine. Now, come to the pharmacovigilance program of India. It was launched by Ministry of Health and Family Welfare, Government of India, in the year 2010 at AIIMS, New Delhi, as a National Coordination Center. The program was recasted at Indian Pharmacopoeia Commission as National Coordination Center in April 2011 by a notification issued by Ministry of Health and Family Welfare, Government of India. IPC-PVI became the National Coordination Center for Material Vigilance Program of India from July 2015, and we are working on that area also. IPC-NCCPI became a WHO Collaborating Center for Pharmacovigilance in Public Health Programs and Regulatory Services from July 2017. So this is the inauguration by the Additional Secretary Dr. R.K. Vats, who inaugurated the WHO Collaborating Center in Indian Pharmacopoeia Commission on the date July 18, 2017. Now, it is a first of its kind. The World Health Organization on July 18, 2017 bestowed upon NCCPVI as WHO Collaborating Center for Pharmacovigilance in Public Health Programs and Regulatory Services in India. So, come to the organogram of Pharmacovigilance Program of India. It is administratively headed by Secretary, ex officio Ministry of Health and Family Welfare, because it is the in charge of governing body and scientific body and general body of Indian Pharmacopoeia Commission. Again, the program is being administered by Secretary cum Scientific Director, Indian Pharmacopoeia Commission, and it is equally supported by the National Scientific Coordinator and Steering Committee, Working Group, Signal Review Panel, Co-Training Panel, and Quality Review Panel. The Steering Committee's work is to give direction to the program in an administrative way. Working Group give the direction regarding technical inputs into the program, like extension of the program and any recommendation that we need to give to the CDSO. It's went through Working Group. Next is a Signal Review Panel and Co-Training Panel and Quality Review Panel. Quality Review Panel is responsible for the quality of ICSR and completeness of ICSR. Co-Training Panel is responsible to for the establishing the various modules for different kind of participants. Say, for example, from doctors, pharmacy students, and nursing staff. So we have different kind of modules in pharmacovigilance for the different people. So these all modules are being designed by Co-Training Panel. Next is the Signal Review Panel. Signal Review Panel has the responsibility of identifying signal and any outcome from the analysis from the ADR database. Further, when we come to the hierarchy, it's come to the Senior Principal Scientific Officer, who is the program in charge, or you can say the officer in charge for the pharmacovigilance program of India, and also responsible for the quality management system and drug safety management system for the pharmacovigilance program of India. It is equally supported by senior scientific officer and scientific assistants here, and there is a large team of pharmacovigilance associates who are working at National Coordination Center as well as at Adverse Drug Monitoring Centers. Here, supporting divisions of pharmacovigilance program of India, we have human resource, administrative divisions, finance and accounts division, information technology divisions, store and procurement divisions. All the logistics supports for the program is being provided and human resources provided through these divisions. Next, come to the ADR monitoring centers. ADR monitoring centers are being, as you know, that at present we are having 311 radiomedic center across the India, and it is being managed by a team of pharmacovigilance associate at National Coordination Center. There is almost 30 pharmacovigilance associates are working at National Coordination Center, and they are responsible for a group of AMCs for giving them technical inputs regarding the ICSR management. And for ICSR management, we are using a software provided by WHO UMC, and that is VisiFlow, and we are working with that software. Apart from that, we are having in the drug safety management system various divisions like ICSR processing, pharmacovigilance regulatory assessment, signal detection, benefit assessment team, national health program team, research and development team, and international cooperation teams. So ICSR processing division is almost all pharmacovigilance associated at National Coordination Center are looking after this ICSR processing at AMCs also. I'll show the end-to-end ICSR flow in my upcoming slides. PV regulatory assessment is a division which is responsible for coordination with the marketing authorization holder. As you know that in 2018, we have given a guideline for the marketing authorization holder for establishment of pharmacovigilance system for their medicinal products. And that guideline also gives some brief about that guideline also. Next, we have signal detection team. Signal detection team is responsible for receiving the inputs from the AMCs also, and from AMCs it go to the signal detection team, in-house team of PVPI. They analyze the data. After confirmation at the ground level, at the signal detection team level, we compile the data and put forward to the signal review panel here. Then signal review panel analyze the data and give recommendation to the working group, and through working group it's going to CDS. Next, we have benefit risk assessment team also. It is also looking after this. It is also supporting the signal detection team. We have national health programs. As you know, as a National Coordination Center, we have collaboration with various national health programs like National Tuberculosis Elimination Program and National Vector-Borne Disease Control Program, National AIDS Control Program, and Universal Immunization Program. So we are attached with these programs, and we are getting data from these programs also. Next, we have research and development divisions, those who are responsible for doing the research in the data and the research in the database, and the persons at the coordinator at the adverse drug monitoring centers also sending some inputs to the research and development divisions, and they are working very nice. Next, we have international cooperation division, which is responsible for to collaborate with the international organizations. Like recently, we have some interaction with the regarding pharmacovigilance with Ghana from South African country like Ghana and Bhutan. They visited our National Coordination Center, and we given a training and sometimes few times back say from certain few candidates from Ethiopia. They also visited us. I'm sorry. Next, we have quality management system. Basically, I'm the person who's looking after these divisions. Quality assurance, quality control. It is based on the ISO 9001:2015 guideline, and we have established all the standard operating procedures for doing for the mandatory activities of the National Coordination Center. Next, we have training and education division, which work in coordination with coordinating panel. Next, we have promotion, communication, and publication division, which is basically responsible for the publication of resource material, and they are also responsible for coordination with the regarding various articles to be published in various journals through the AMCs or NCC. So next, come to the pharmacovigilance in acts and rules. As you know that pharmacovigilance is mentioned in Schedule M of Drug and Cosmetic Act, para number 28, sub para 2. It is mentioned that all the serious adverse drug reactions, the data related to all the serious adverse drug reactions.
This is Sarada.
Yes, ma'am. Yeah. Hello. Am I audible? Hello.
Yes, sir, you are audible, sir.
Yeah. So pharmacovigilance in acts and rules, as you know that Schedule M of Drug and Cosmetic Act, it is mentioned there in para 28 subpara 2 that all the serious adverse drug reactions need to be forwarded to the concerned licensing authority. The timelines have not been mentioned there, but in new drugs and clinical trial rules 2019, it has come. They have given some timeline also for submitting the data. The fifth schedule explains about the requirement of pharmacovigilance, that it should have some pharmacovigilance system for monitoring the safety of medicines in the post-marketing phase. Next, Pharmacy Council of India pharmacovigilance as one of the subjects has been included for pharmacy undergraduate curriculum. And recently, in National Health Policy, pharmacovigilance has found some space for the audit, prescription audit inclusive of antibiotic uses. It was released by Ministry of Health and Family Welfare, Government of India. Next is it is the distribution of our ADR monitoring center. We have 133 non-government ADR monitoring centers and 178 government hospitals. We have total 128 pharmacovigilance associate at different AMCs and NCC. These are the number of ADR monitoring centers in state-wise. PPI collaboration, as I told you, that we have collaboration with various national health programs. Currently, we have 21 area monitoring center in under national tuberculosis and elimination programs, and 17 center under National AIDS Control Organization. These 17 centers, few of them are center of excellence for treatment of AIDS. And next, we have 58 national vector-borne disease control program centers under our flag. And we are looking for the adverse drug reaction data related to Kala-Azar, the drugs which are used for the treatment of Kala-Azar. Next, we have the collaboration with Universal Immunization Program. The events following events following immunization, we are getting information, this area-related information regarding immunization through this network of AFI. This is the structure level, that is at level 1 we are National Coordination Center. Level 2 we have regional training centers, as it is rightly mentioned that JSS College of Pharmacy is one of the regional training center for our South Zone. And next at the next level we have AMCs under the different regional training centers. These are the number of ADR monitoring centers that we grow year by year. In 2012, we have 86 number of ADR monitoring centers, and subsequently in the years we are increasing. And I would like to share with you that at the end of 2020, maybe at the end of December 2020, we are going to include 40 more number of ADR monitoring centers in our lab, and at the end of 2020, we will have 350 ADR monitoring center under pharmacovigilance program. We are analyzing the data. We are receiving letter of intent. It is being analyzed by our team, and subsequently will be approved by working. So PPI data sources. We are receiving the data from ADR monitoring center, marketing authorization holders, national health programs, and through patient. That as you know that we have options for the healthcare professionals and consumers to directly report to NCC. We are also receiving reports from patient and consumers. This is the slides basically for the end-to-end ICSR flow from AMCs in PVPI. How AMCs are reporting, that I try to conceptualize through this particular slide. Like adverse drug reactions is reported by reporter at AMC or NCC, data processing at AMC at NCC. Next, quality assessment at NCC through QA division. Now, if the quality factors are met, quality criteria, then it is being forwarded for the clinical relevance to the pharmacovigilance associate responsible for this activity. And if they found any conclusive data to be presented to the signal review panel, then it goes to the signal review panel. Afterwards, if signal review panel recommends, so it goes to the CDSU. Next, if the quality is not met, then it is upgraded at NCC with the help of the reporter, because the information it's a two-way communication between the reporter and the NCC. We collect more information regarding the idea, and then finally we submit the idea to UMC through VisiFlow. These are our PVPI communication channel. Few of the things have already have been explained, except this marketing authorization holder. Currently, we are getting ICSRs from the marketing authorization holder in the form of E2B XML format, and we are processing through these ICSR through VisiFlow and sending it to the VGBase. Next, a little bit introduction about the pharmacovigilance guidance document for marketing authorization holder. Holder of pharmaceutical products. It's basically made up of 6 modules. Module 1 give some direction regarding establishment of pharmacovigilance system master file. Module 2 give collection, processing and reporting of individual case safety reports. Module 3 gives direction about preparation and submission of PSUR. Module 4 quality management system and marketing authorization holder organization. Module 5 give audit and inspection of pharmacovigilance system at marketing authorization holder organization. Module 6, submission of risk management plan. This particular document is available on this on the website of Indian Pharmacopoeia Commission and also available on the website of CDSU. So anybody, if any student or any person would like to go through this particular document, they can download this document and study it. It is very good for those persons who want to make a career in pharmacovigilance in pharmaceutical industry. Next, it is the data that we have received year by year, and as on date, we have 4,64,087 ICSR in our database, and it is continuously increasing. Because of COVID-19, there is a very less number of, as compared to previous year, less number of ICSR there, because during COVID-19 we received less data, but we are recovering now, and ICSR is being submitted by the various area managers to PVP. This is regarding the completeness score. As you see that in the last 2 phases, the completeness score of India is getting down. This is because that we were having the more number of reports from the pharmaceutical industries. As you know that quality and completeness is a very big cause of concentration, that the quality we are receiving from marketing authorization holder is not good. And in this month, in this quarter, we have uploaded maximum number of reports from marketing authorization holders, because all the PV associate through various EMCs, we have large backup for the ICSRs. So we uploaded all the ICSR in the VisiFlow, and that is the reason that our completeness scores getting down in this quarter. And it is in the phase of recovery, and hopefully, apart from that also, we are much ahead with other countries. As you say that all other countries' quality score is quite low as compared to India score, and hopefully we will improve from this situation. Next slide. So these are our ADR reporting form. Red color form is for healthcare professionals, and blue color form for consumers. And consumer reporting form is available in 10 regional languages like Hindi, Gujarati, Kannada, Bengali, Malayalam, Oriya, Tamil, Marathi, Telugu, and Assamese. And we are all under few other languages form is under process. Soon we will have one consumer reporting form in Urdu language. During COVID-19 pandemic, National Coordination Center has designed a suspected adverse drug reaction reporting form for drugs used in prophylaxis treatment of COVID-19. Specifically, because we have received a direction from the Ministry of Health and from CDSU for the focused pharmacovigilance for the drugs being used for COVID-19. And we are, through our ADR monitoring center, we are receiving such reports, and the analysis of these reports is being regularly submitted to Government of India and CDSU on a regular interval for any regulatory decision. I would like to explain here that recently we came out with some unlisted ADRs regarding hydroxychloroquine, and in the upcoming meeting we are going to discuss that event with our signal review panel, and hopefully in the next couple of days it will come as a signal for hydroxychloroquine. The channel of reporting and adverse drug reaction in PVPI, as you know that ADR PVPI app is freely available on the Google Play Store. You can download it, and it is very easy to give information regarding ADR PVPI for intellectual people who are using a smartphone. For the all peoples, as well as healthcare professional and consumer can report through ADR PVPI, and it is a paperless, seamless transfer of ADR related information to the National Coordination Center or the ADR Monitoring Center. It gives you option of selecting the AMC as well as NCC. Apart from that, you can report adverse drug reaction to our helpline number on Monday to Friday from 9 a.m. to 9:30, and the number is 1800-180-3024. You can remember this number by 30 into 24 hours. You can say it's a kind of mnemonic that I suggest. It is easy to remember this. Next, we have ADR reporting form that I already explained. Next, we have ADR Monitoring Center, 311 ADR Monitoring Center. Through this, the ADR adverse drug reactions can be reported. Underreporting a major concern, because people are very worried about few of the causes mentioned in this particular slide, like afraid of legal liability, medication errors, off-label use, one additional administrative burden. I'm not paid to report ADRs like that. How will my one report help? I don't know what to report, all or some. I don't have time to fill out the ADR reporting form. So these are all the excuses that we used to face from healthcare professionals being a pharmacovigilance day. But after all, we being a healthcare professional, it is our moral responsibility to report adverse reactions that we came across. So these are the PVPI resource materials, performance report. NCC PVPI is regularly publishing newsletters and on every quarter, and guidance documents, and this pharmacovigilance guidance document from MEH, and quality manual. Quality manual is a basic documents for establishing QMS at NCC PVPI. Public outreach. Till date, we have conducted 3,058 training and sensitization events at various ADR monitoring center and at National Coordination Center. We have trained 134,188 healthcare professionals in pharmacovigilance. We have broadcast 8 programs on TV and radios. We have given SMS facilities available with us. There's drug alert. Near about 11,215 SMS already have been sent to various healthcare professionals. These are our publications which highlighted in the WHO newsletter. Also, you can go through. It is available on the website of WHO. Next, these are some of the clippings, recommendation of a signal review panel that has been captured by various media, print media. These are the outcome of our pharmacovigilance program of India. Till date, we have released 6 signals like cefixime, AGEP, itraconazole, furosemide dermatitis lichenoids, lithium carbonate, drug rest with eosinophilia and systemic symptoms, fluconazole, hyperpigmentation, oseltamivir, sinus bradycardia. It is under consideration at CDSU. Other regulatory action by signal panels are drug alerts. We have released 102 drug alerts, and we have recommended some package insert updating 55. It is number in 55, and this information is also available at IPC website as well as CDSU website. So these are our Facebook and Twitter accounts, and through which anybody who are interested in reporting ADR or to understand the pharmacovigilance in pharmacovigilance program of India, they can visit these websites, and these options they can follow us on. So that's all from my side. Thank you very much. Dr. Roji?
Thank you so much, sir. Thank you so much for giving valuable insights on topic recent update on pharmacovigilance program of India perspective. So we have a few questions from delegates. So one of the delegate named Aishwarya Babu, she's asking that how can I work in pharmacovigilance abroad?
Abroad?
India, both.
For abroad, you can work through various pharmaceutical industries. You need to give some interviews. You need to visit their website, and if some vacancies are available there, you can apply there. But before application, you should have some experience in pharmacovigilance. You can have experience through various training programs also, and try to get some. If you have completed your education, you can have do pharmacovigilance job in India, and afterwards you can apply for abroad.
So there's another question. It's relevantly same. So how to be part of PVPI team as a contributor or a consultant?
Yeah, being a contributor is anybody, being a consumer, being a healthcare professionals, they can send adverse reaction to PVPI through email ID, through helpline, through ADR PVPI app, Android mobile application. And for to be a part of PVPI team, you need to apply through various vacancies. As you know that recently, today also we are conducting interview for pharmacovigilance associate post. We have released near about 147 pharmacovigilance associate posts in the last month, and the interview for that post is going on.
Thank you, sir. Thank you so much, and thank you for sparing your valuable time and giving this very informative session. Thank you, sir.
Thank you. Thank you all the dignitaries for making me part of this wonderful platform.
Thank you, sir. Thank you. So with that, we will go ahead for our next speaker, Dr. A. Pramod Kumar, Associate Professor, Department of Pharmacy Practice, Chebrolu Hanumaya Institute of Pharmaceutical Sciences, Guntur, Andhra Pradesh. I'm privileged to introduce our next speaker, Dr. Pramod Kumar. So Dr. Pramod Kumar has completed MPharm and PhD from JSS College of Pharmacy, Mysuru. He's a pharmacovigilance expert overall with 5 years of experience, and he has been previously worked for the National Coordination Center Pharmacovigilance Program of India, Indian Pharmacopoeia Commission in Ghaziabad, and he has been expertised in sensitization of healthcare professionals on pharmacovigilance, establishing a pharmacovigilance setup in hospitals, signal detection, and literature search. So he has published more than 30 papers in peer-reviewed journals and has attended 55 national and international conferences. And he is currently working as associate professor at Chebrolu Hanumaya Institute of Pharmaceutical Sciences at Bengaluru. And he is also the deputy coordinator for MD-AMC, Associate MI-NRI College and Hospital, Bengaluru. Now I request Dr. A. Pramod Kumar to deliver the time. Over to you, sir.
Yeah, thank you, Rosie, for that nice introduction. So before I start the presentation, let me thank Kidwai Institute, Sarada Vilas College of Pharmacy, Department of Clinical Pharmacy, JSS Academy for organizing this wonderful program and giving me opportunity to interact with you all. And also I especially thank Dr. Hanvanta Joshi sir and his team for arranging this without any hassles and conducting a smooth program. So I am sharing my screen. Let me know once you are able to see that. So is my screen visible?
Yes, sir. So you can share the screen, sir, if it's not getting uploaded.
Yeah, I have shared it again.
Right. So it is not yet visible. Yes.
Is it clear now?
Yes, sir. It's visible.
Yeah. So I'll be presenting on the topic detection and reporting of adverse drug reactions. So indeed, it's a pleasure for me to share this platform because there are some mentors of mine like Professor M. Ramesh, Dr. Juni and all. So I take this as a pleasure for sharing this platform along with them in sharing this information. So with this, I'm entering into the session. So this is my disclaimer. So the views and opinions expressed here are individual and not affiliated to any of my previous organizations or to the current organizations or for the organization team. And I don't have any conflicts of interest in presenting this data. So coming to the history, as we all know, the pharmacovigilance has initiated in the year 1962 in US, and it took almost 4 to 5 decades for coming into the developed part of the world, including India. But we can say it is still in infancy stage in India, and lot of amendments has to be taken place in order to go ahead in the field of pharmacovigilance. As we all know, adverse drug reactions are leading cause of death throughout the world, because according to the statistics we can see it stands in top 10 of the mortality causes globally. Here we know the polypharmacy is acting as a significant factor. So one published data, if you see, that is Indian Journal of Medical Research, wherein they have assessed the risk factors for the adverse drug reaction in Indian ambulatory elderly patients. So if you see the risk factors in that study, I have shown here that the age and the number of medications increasing, so the number of adverse drug reactions are also increasing. So with this, it is very much evident that because of the increase in age, as the comorbidity conditions increases, wherein the patients are needed to take more than 5 to 6 medications, at that time they are more prone for this occurrence of the adverse drug reactions. Hence, whenever there is a polypharmacy, we need to be more vigilant towards the identification of adverse drug reactions as well as its management. So coming to the safe drug, as we all know, no drug is entirely safe. Each drug is associated with some or the other adverse drug reactions or the side effects. So there are the risk in intrusion into the human body, whether it is a chemical, surgical or any kind of procedures. So we need to be more vigilant when we are using this type of medications or any procedures in the medical field. So why so many adverse drug reactions are occurring here? The first reason is because of the use of large number of prescriptions. As we all know, there are some irrational prescribing also which is going on, and unnecessarily prescriptions are being given to the patients. Hence, that is the first reason for occurrence of more number of adverse drug reactions. And recently we have seen in the previous slide about the multiple drug therapy, which is a significant risk factor. Next is lack of proper system to detect and monitor the adverse drug reactions. As you have seen in the previous presentation that Mr. Rishi Kumar has presented, that we have only 311 adverse drug reaction monitoring centers till now. When compared to the entire country, this number is very small. So still we need to strengthen our reporting system, and we need to frequently monitor and detect them in order to have proper presentations. So that is why we can say there is a lack of proper system. Not only in our country, but if you see in all developing countries, this is in the same scenario. So once we lack the reporting system, there are many adverse drug reactions which are not reported frequently to the other healthcare professionals, so that the awareness will not come then and they cannot identify the adverse drug reactions. So this is the reasons. So you will get doubt why there is a need to monitor the adverse drug reactions in the already marketed drugs, because it passes through all the clinical trials. So what is the need again to do these things? But if you know that there are some major limitations during these pre-marketing safety studies. So when we are conducting these clinical trials, we will not include the patients who are having impairment in the liver and kidney diseases. And it is limited information about the drug effects in specific ethnic groups, because whenever you are conducting the clinical trials, there is no rule to include all the ethnic groups into the study. Only we will go with the number of the patients. Hence, we will be missing the data to include the various ethnic groups that is present in the worldwide. Also, there will be a limited information about the drug-drug or drug-food interactions, because when we are doing these clinical trials, we will not be exposing our molecule to all the other drugs or with the various foods that are available. Hence, once it is marketed, it gets exposed to n number of drugs and the foods, even to the herbals also. So the number of interactions will be increasing, and there will be chances of getting more adverse drug reactions. And also, during the clinical trials, we will not be missing to report the long-term adverse effects. Because of the time constraints that we have during the clinical trials, we will not be monitoring the long-term adverse drug reactions that can occur after 10 to 15 years of usage of the medication. So all these are the limitations which will be missing during the clinical trials. Hence, after post-marketing also, we need to be completely monitor the safety of the products. And one more important is, as you all know, in clinical trials, we will not be including some special populations such as elderly or some pediatric cases, and also pregnant and lactating women, if the drug is not intended for them. But once it is marketed, suppose the new molecule has come for epilepsy, during the trials we will not be including any pregnant women because of the known teratogenic effects. But once it is marketed, maybe because of the benefit risk ratio, the pregnant women may need to take that medication. So once they take that, you can see in this slide what are the various effects that various class of drugs can show as and classified as the teratogens. If you see the anti-epileptics, commonly we can observe in the newborn is the cleft palate and neural tube defects. And if you see some cytotoxic drugs, almost all are presumed to be very severe teratogens. And apart from that, if you see commonly used ACE inhibitors and all, it can lead to oligohydramnios. So all these are the examples that each class can affect the population once it is into the market. So what pharmacovigilance includes? So pharmacovigilance is not restricted only to reporting of the side effects, but it has extended its scope. Now it is not limited to only of reporting the side effects or the adverse reactions, but also you need to monitor properly for medication errors, lack of efficacy reports, abuse and misuse, drug interactions, all includes in the pharmacovigilance. So you'll get doubt what are the types of medication errors. So broadly it is classified as prescribing errors, dispensing and preparation errors, administration errors, monitoring and dose adjustment errors. So under which you have different types of things like wrong patient, wrong medication, or wrong formulation, wrong route has been prescribed, or the patient has taken in wrong route. Known medication allergy means the patient is already allergic to some medication and the same class of medication was given again. So that is the known medication. So all these are the different types of medication errors. If you see, the prescribing errors arises from the physician side, wherein the prescriber is giving the medications, and dispensing and preparation errors can occur at the compounding level from the pharmacist. Administration errors we can see from the patient side or consumer side or from the nursing staff side, and monitoring and dose adjustment errors again comes from physician and pharmacist together. So all are at different levels, these type of medication errors do occur. You can see the study published in Journal of Pharmacology and Pharmacotherapeutics. So they have seen the prescription errors in cancer chemotherapy. So in that, the omissions has superseded the potential harmful errors. Means the physicians or prescribers, whoever has given that medications, has some omissions in the prescription. So this omissions has superseded among all the harmful errors that has occurred during the chemotherapy management. So in that omission, you can see some people has missed the dosage form, some has missed the units of dosage, and missing information on diluent to be used, missing information on route of administration, whether it is IV, IM, or subcutaneous or not. So there are different types of omission errors. So this is one type of table that has been published in the year 2015. So it is available on free access. So you can also Google that with this PM ID, you can note down. So like this, there are different studies wherein it can show what are the different type of medication errors that are happening in the cancer chemotherapy hospitals. So after the medication errors, we can see the adverse drug interactions. So this drug-drug interactions is accounting for 6 to 30% of all the adverse drug reactions that are occurring in the global level. So if you see the frequency of potential drug-drug interaction out of this, it is up to 4 to 46% according to one more study. So basically what you need to understand here is there are some hospital admissions which are occurring because of this drug-drug interactions. So once we know properly about this drug-drug interaction, we can try preventing this from the hospital admissions. And also it is not because of the hospital admissions. At many times it will increase the length of the hospital stay because of administration of these drugs, which leads to the drug-drug interactions. So you can see here the examples of adverse drug interactions. So anticoagulants, very well you all know, giving of warfarin along with enoxaparin leads to increased bleeding. So immunomodulatory agent cyclosporin, when given with antilipidemic simvastatin, leads to rhabdomyolysis, that is muscle damage. So when we use antiplatelet like aspirin, clopidogrel, and same here we can see the increase in bleeding condition. So SSRIs like sertraline, when given with olanzapine, we can see increase in the weight in the patients. So all these are the examples of adverse drug reactions. So for the same drugs, I have taken the interaction with one of the chemotherapeutic agent, doxorubicin. Here you can see warfarin, when we give with doxorubicin, it increases the risk of bleedings again. And when we give cyclosporin along with doxorubicin, it increases the toxicity of the doxorubicin. And when we give with the olanzapine, it leads to irregular heartbeats. Whereas with aspirin and clopidogrel, there is no interaction. So if you see randomly, if we have taken 4 drugs, out of these 4, for 3 drugs, we can see the interaction. So that is how there is a potential for chemotherapeutic agents to have more adverse drug interactions. Hence, we need to very more properly monitor whenever it comes to the polypharmacy in cancer patients. It is not limited to only the chemotherapeutic agent with other drugs. So within same class also, among the various drugs of chemotherapeutic drugs, there will be some adverse drug interactions. So other examples where we need to keep in mind when we are prescribing these chemotherapeutic agents are, first is any live vaccine we should not give. For example, I have quoted here about the rotavirus vaccine. So concurrent use of any live vaccines along with chemotherapeutic agents is completely contraindicated because it leads to increase in the infection with which we are giving the vaccination. So likewise, as you know, though it is not a direct interaction, but the absorption will be changing to capecitabine and lapatinib when we are giving along with the food. Hence, it is recommended to take before or after 1 hour of food along with the water. Next, if you see, there is one class called tyrosine kinase inhibitor. So its solubility and bioavailability changes with increase in the pH. So when we are giving this tyrosine kinase inhibitors along with any acid suppressive agents, the bioavailability will be changing because the tyrosine kinase inhibitors needs the low pH for better action. So except imatinib, all tyrosine kinase inhibitors should not be given with acid suppressing agents, wherein there is a potential adverse drug interaction that can take place and the bioavailability of these kinase inhibitors will be reducing. So these are the various examples that we have for the adverse drug interactions. So likewise, we have a long list. You can just go and surf in the Google that what are the aspects that we need to keep in mind when we are going to prescribe chemotherapeutic agents along with the other drugs. So also, you know, there are some drugs that have been banned. Those are called fixed dose combinations. Means these combinations should not be used. So recently, 2 years back, there are a long list of drugs like around 350 fixed dose combinations has been banned by our Indian regulator, that is CDSCO, Central Drug Standard Control Organization. So here I am quoting few examples of it, like tetracyclines along with vitamin C, we should not be prescribing. Fixed dose combinations of antihistaminic with antidiarrheals should not be given. And fixed dose combination of laxatives or antispasmodic drugs should not be given. So these formulation kind of things are completely banned. So pharmacovigilance is again having some transition. So apart from the various things that we have seen in the previous slides, it is now moving into the advancements, like it is entering into the pharmacoepidemiology, wherein we need to see in the set of population how the drug is acting on them. It can depend on the disease condition, it can depend on the region and ethnic group. So that is about the pharmacoepidemiology. When it comes to the pharmacogenomics, it is a role of genome in drug response. And when it comes to toxicogenomics, it is collection of information about gene and protein activity in a cell in response to toxic substances. Whereas in pharmacoeconomics, you can see the best model according to the economics and which is cheaper among the two drugs when we compare the drug A and drug B. So likewise, various comparisons has been taken place in order to give better choice of treatment to the patients so that it will not have any burden economically on them. So like this, the pharmacovigilance has been advancing on a very rapid pace. So these are the new fields which you can look into, like pharmacogenomics, toxicogenomics, and the other areas wherein the research is taking place. So how it helps like advancement in these fields is like here you can see the predictive pharmacovigilance. There is a strong association between carbamazepine and occurrence of Stevens-Johnson syndrome. So what is the association is the presence of HLA-B1 1502 allele will cause this type of reactions in the patients. So if you see all the patients who have suffered with SGS after taking of carbamazepine, they are identified with this HLA-B1502 allele. So likewise, once we understood the genomics, there can be some predictive pharmacovigilance we can take and we can prevent the occurrence of that adverse drug reactions. So likewise, one more example, when there is a variation in CYP2C9, it can alter the anticoagulation response from the warfarin even we give it the normal dose. So that is how the adverse drug reactions do occur because of the change in the genome content. So based on that only the drug will be showing its response. So where you have to report, in the previous session you have seen that we have the National Center to collect all the adverse drug reactions which are occurring in our country. Next is when. So as an academicians, you can report at any point of time. It is not restricted to any time period. Only for the industry perspective, we have some limited time periods. How you can directly report it through email or through telephonic. What to report? It is not that only serious reactions or the unlisted reactions you have to report. So initially you have developed that habit of reporting even if it is a non-serious and even if it is a listed. Like many of the healthcare professionals will be asking us, what is the need of reporting this listed? When we know that chemotherapy will cause the nausea and vomiting, still what is the need to report that reactions? But it is not like that. Even if you know the drug will cause certain reactions, you need to report that reactions to understand about the frequency of the or incidence of that reactions in that particular country or in that particular locality. So in order to understand that, we need to still report even if it is a listed or non-serious reaction. Who can report? Any healthcare professional who is a nursing staff, pharmacist or physician can report these reactions. Even consumer can report these reactions. That's why PVPI has come up with reporting form that is exclusive for the consumers. Why to report? So in the entire session, all the speakers will be speaking on this, why we are doing this and what is the importance of conducting this pharmacovigilance. So many healthcare professionals ask, so is it really important to report the adverse drug reaction so that the National Center will take some actions? Yes, definitely. So in this slide, you can see what are the drugs that have been withdrawn because of the adverse drug reactions. So antihistamine terfenadine is leading to severe ventricular arrhythmias. So that's why it has been banned in our country. Stilbestrol, which is a non-steroidal estrogen leading to carcinogenesis, has been withdrawn from the market. Practolol, which is a selective beta blocker causing oculomucocutaneous syndromes, wherein the fibrosis of the retina occurs. Nimesulide, yes, it is only withdrawn in use of pediatric population, but not in adults. Servistatin causing rhabdomyolysis. COX-2 inhibitors causing severe cerebrovascular events. Rimonabant, like causing psychiatric disorders, which is an anti-obesity drug. Tegaserod causing severe cardiovascular effects. Gatiproxin, yes, systemic use is causing fatality, hence it has been withdrawn. Only some eye drops or such kind of things are being used. Mepacrine causing ectopic pregnancy and leading to carcinomas. Methaqualone, which is a similar class of chloroquine, causing drug abuse. Nialamide causing hepatotoxicity, which is an irreversible monoamino oxidase inhibitor. So all these are some of the examples, because it is leading to severe adverse drug reactions within short period of time after coming into the market. Hence, it was brought into the notice of the Indian drug regulator and it has been subsequently withdrawn from the markets. You can see here, this is a very good example. What is the need for withdrawing into the Indian market, like pioglitazone. So pioglitazone has been banned in the year 2013, I think, in the month of June. So because it has been withdrawn in many countries of the European region. So by seeing that, Indian government has also suspended initially. So the drug manufacturers has filed a case because there is no data that pioglitazone is causing risk of or having risk of bladder cancer in Indian population. How can you ban this drug? So the court has again given the orders to revoke that and the manufacturers can sell this drug, because what is the main point here is the manufacturers has went to the court that there is no case in India that pioglitazone has caused bladder cancer, then why we need to withdraw it. So that is the importance of reporting the adverse drug reactions. Even at your hospital, if it is one case, you should not think that it will not having any significance. Even one case will help in such conditions in maintaining the health of our Indian public. Later, many studies have been published. So one of the study from the BMJ, you can see pioglitazone use and risk of bladder cancer, which is a population-based cohort study conducted in UK. Like that, many studies have been there. You can see and what are the countries that has been banned the use of pioglitazone. So like this, we have 3 to 4 examples wherein the suspension has been done and later they have been revoked into the market like that. So what are the factors that contribute to these adverse drug reactions are age, race, gender, drug-drug interactions, drug-disease interactions, drug-food interactions, drug-herb interactions, and genetic factors, which we have seen in the previous slide. With the increase in number of these things in a patient, the risk will be more for developing the adverse drug reactions. So integration of pharmacovigilance program that has been already covered in the previous session, like now PPI has engaged with all the AEFI, adverse events following immunization from the Immunization Technical Support Unit ITSU, and also with revised National Tuberculosis Control Program, National AIDS Control Program, and National Vector-Borne Disease Control Programs and all. So wherein we can do the targeted pharmacovigilance. So patient is having HIV and also the patient is suffering from cancer. So when the patient will be using antiretroviral drugs along with the chemotherapeutic agents. So at the time there are a lot of scope to conduct this targeted vigilance to see various drug interactions that are occurring in that specific group, and also what are the different level of pharmacogenomics that is happening, which helps in giving the evidence-based data which can be applied to the another set of population. So now you will be interested to report the adverse drug reaction because you have understood what is the importance of it and what are the types of adverse drug reactions. Now you will get doubt where you need to look for the information whether this drug will cause this reaction or not. So in order to understand more about the pharmacovigilance, you have many information resources like Uppsala Monitoring Center and all, wherein many YouTube links are being available, which are free of cost. You can just go into YouTube and just see about this pharmacovigilance programs. There are some journals which are specific interest. If you want to understand about the pharmacoepidemiology, you can go to Pharmacoepidemiology and Drug Safety specific journal. Like that, we have some special interest journals, which I have quoted here. You can note down and you can go into these journals. And when it comes to oncology journals, we can go to Annals of Oncology, Journal of Clinical Oncology, Journal of National Cancer Institute, Cancer Treatment Reviews, Cancer and Metastasis Reviews. So like this, various oncology specific journals are there, wherein you can get all the latest information about the drugs which are used in these cancers. Apart from that, you have some reference books like Myler's side effects. So wherein the side effects of each drug has been illustrated in a very descriptive manner. Like we have reference books Martindale, Pharmacovigilance from A to Z, written by Lobot. You have some textbooks like Side Effects of Medical Cancer Therapy, published by Spingers, Recent Results in Cancer Research, Textbook of Pharmacovigilance by Elizabeth Andrews. So all these are various textbooks that gives you more information towards the pharmacovigilance and especially with respect to medical cancer therapy. And if you want some addressing to the specific problem, reference books like what are the adverse drug reaction that has been known to occur in the pregnant and lactating women. In that case, you can directly go to this Briggs-Freeman reference book. Like that, if you want to see only the ocular side effects, you can go to the Frauenfelder's book. And if you want to understand about the adverse effects to the herbal drugs, you can go to Dismet et al. Books. So all this has been updated very much recently. So what I'm showing is the older versions. So you can see these reference books, you can procure them and you can use these for better understanding of all the pharmacovigilance. So apart from the journals and the textbooks or reference books that you have, if you feel that it is more costlier, then always you can go with the computerized literature that is available free of cost in many of the times. Like PubMed, you can go, that is a Embase, Medline. So there are various data sources that are available for you in online, wherein you can go and read all this type of articles that are present. Apart from that, each country is having some national adverse drug reaction bulletins, but it is not specific to only chemotherapy. You can see wider class of drugs will be covered here. And there are some non-governmental sites where you can get the information about the adverse drug reactions. You can also go through that, like Uppsala Monitoring Center, Center for Education and Research on Therapeutics, CERT, like that. Many NGO sites are available for you. So all these are the acting as sources for you to gather information, and then you can report the adverse drug reactions. So as I said, reporting form are two types. The red colored form given by Pharmacovigilance Program of India is for healthcare professionals, and the blue colored form is for the consumers. So the only intention is the consumers will know less information about the drugs that they are consuming. So it will have some less fields compared to the healthcare professional form. So the main aspects of this form are patient information, suspected adverse drug reaction, suspected medication, and reported details. So these are the main 4 sections that you need to include when you are reporting any type of reactions. So in patient information, you need to collect the gender, weight, and age of the patient during the time of event. So patient's name is not required. You can just enter his initials in order to maintain the confidentiality. Suspected adverse drug reaction information, you can just write about the date of reaction that has started and when it has been recovered or when it is still recovering or not, and describe the reaction or problem. So each one will be including some more data. So you need to have some short version of that description. So suspected medications, you need to include all the drug details like dose, how much you have used, what is the frequency that has been given to that patient so that the adverse drug reaction has occurred, what is the route that has been taken by the patient, and who are the manufacturers for that. So why it is required to enter the manufacturer detail is whenever we have some series of events, we can directly consult the manufacturer. If there is some discrepancy, they can also recall that batch. That's why the manufacturer and batch number are required whenever you are reporting any adverse drug reactions to national centers. Causality assessment, it is an entirely different session. In the next session, you can see it from Professor M. Ramesh sir. So wherein you can understand more about this causality assessment. So about the suspected medications. So what is the action taken, whether the drug which is responsible for that reaction has been withdrawn or not, the dose has been increased or decreased during the occurrence of that adverse drug reaction has to be mentioned clearly. And if it is not changed also, you have to tick that box. So apart from that, you have to mention always the concomitant medications. So concomitant medications are the drugs that has been taken by the patients at the time of occurrence of event. So it is not only one drug that is suspected, but also when you are review concomitant medications, we can see whether there is some drug interaction that is responsible for occurrence of that adverse drug reaction. So you should not get confused with the management part, because whenever the adverse drug reaction occurs, the physicians will manage the patients by giving some additional product. So you should not write that product in these columns. So you should not get confused whenever you are giving dexamethasone or aprepitant in nausea vomiting patients. You should not give this treatment part at the concomitant medications. So generally the nursing staff will get confused with these treatments. So that's why I have been pulled this data. So because of chemotherapy, we see these reactions like neutropenia, anemia, acneiform rash, and mucositis because of even for the radiology we can see. So for all this, we give treatments like filgrastim, eptoin, clindamycin, and lidocaine and all. So nursing staff generally gets confused and write these drugs at that concomitant medications. So we should be educating them that the medication part should not be entered in this concomitant medications or at the suspected drug reactions place. So that's how we need to differentiate them. Finally, you have to mention the reported details. So once the contact is given, whenever there is some kind of signals or drug alerts, the national center or the regional centers can contact directly to the reporters. So other important sections are also there, but only that 4 sections are considered to be mandatory whenever you are going to report any adverse drug reactions. So finally, what are the aims of this pharmacovigilance is to early detect the unknown safety problems. Detection of increases helps in identification, which leads to increased frequency of the reporting. Identification of risk factors, quantifying the risk, preventing patients from being affected from unnecessarily adverse drug reaction, and for rational use of the medications. So PV helps in attaining all these objectives. So as this, you can see here, the WHO pharmaceutical newsletter, wherein the recommendations given by PVPI to the Indian regulator has been published in the newsletter released by the World Health Organizations. So all the healthcare professionals are commonly asking us that what actions will be taken by PVPI in giving recommendations to CDSCO. So here you can see, once PVPI gets all the data, they will submit that to group of physicians, that is called signal review panel, wherein they will take decision whether that is a signal or whether there is a need for updating the package inserts. So whenever there is a need for updating the package inserts, like here example you can see chloroquine causing toxic epidermal necrolysis, so they will be recommending that. So in the January of this year, they have recommended that to CDSCO. And whenever it is a signal that it is not reported much in the world, so it will be directly reported to the manufacturers. So cefixime causing AGEP, that is called acute generalized exanthemus pustulosis, is not much reported in the worldwide. So this has been recommended by CDSCO to all the manufacturers. So likewise, various actions has been taken by the CDSCO. Hence, it is very, very important being a healthcare professional for reporting all the adverse drug reactions that you encounter during your managements. So what are the challenging factors here for reporting the adverse drug reactions? Are lack of knowledge and awareness about the adverse drug reaction reporting systems. So many of the doctors are willing to report, but they don't know the channel where to report these adverse drug reactions. So that was identified as one of the major challenge or blockade for the program. So that's why all these awareness programs are conducted. Next is uncertainty regarding the causality relationship between the drug and the adverse event. So it is not that you need to be very much sure before reporting any adverse drug reaction. Even if you suspect, you can report directly this adverse drug reaction. You should not be 100% sure that this event has been occurred by this drug or not. Next is lack of time or high workload. This we generally see in the government sector because of the high burden. So they are not having proper time to report all these adverse drug reactions. Lack of understanding regarding the importance of adverse drug reaction reporting. Complacency, believing that serious adverse drug reactions are well documented. Lethargy among the healthcare professionals. And fear of medico-legal consequences. So initially we have seen that many of the physicians has this fear of medico-legal consequences, whether if there is a severe adverse drug reaction that is happening in the patient, whether it leads to any complication or the legal issues. It is very much evident, and it has been given by the CDSCO that no physician will be kept under any consequences for reporting any adverse drug reaction. Because being a healthcare professional, you understood that any adverse drug reaction shown by the patient is not pertaining to only the physician fault. It has been various reasons for occurrence of all these adverse drug reactions. So unless you are reporting or giving any wrong medication of wrong route or wrong frequency, you don't have to get fear of reporting any of the adverse drug reactions. In order to conclude my session in respect to time, so adverse drug reactions and PV remains a challenging clinical and scientific discipline always. It continues to expand its domain as we have seen how it is advancing. The benefits of drug therapy should always be greater than the risk. So whenever you are going to prescribe any medication, you have to keep in mind that the benefit should be outweighing the risk that are associated with the use of that medication. So you need to always keep in mind to reduce or minimize the risk by ensuring the good quality, safe, efficacious, and rational use of medications. Finally, knowledge, education, and risk management strategies has to be implemented in all the teaching hospitals and all the healthcare professionals who are willing to be part of this national cause. So the main take-home message for this entire presentation is that Only prescribing, dispensing, and administration of drugs is not only the primary responsibility for all these healthcare professionals, but you need to have and take measures for safe and efficacious use of drugs among our public in the country. So all the audience are most welcome to promote the patient safety in our country. You can join with this toll-free number and the email IDs that has been mentioned. And also you can directly contact the regional centers that has been available, apart from the social media channels that are present by the PVP. Thank you all.
Thank you, sir. Thank you for giving valuable insights on topic detection, reporting, as well as monitoring of adverse drug reactions. Thank you so much. It was a very informative session, sir. So there's a question from a delegate named Jisna.
Okay.
She has asked that how do we get confirmation after we submit the ADR forms to know that our submission has been received correctly?
So whenever you report any adverse drug reactions to the regional centers as well as to the national centers, they will acknowledge that email or the telephonic call. So definitely we'll get some unique ID also once it is entered into the database. So likewise, a series of acknowledgements will be provided by the receivers.
Another question, sir. Which areas need to be reported? Like, for example, nausea, vomiting. So they are a common adverse effects. So which occurs to most of the patients. So whether these common type of adverse reactions can be reported, or should they need to report only the serious adverse events should be reported or not?
Yeah, in one of the slides I have already presented that. So it is not that only the serious or unlisted you have to report. So there is very much important even to report the non-serious adverse drug reactions. Why? Because, for example, all of you might be aware of using the anti-tubercular therapy. So because isoniazid will cause peripheral neuropathy, we give prophylactic treatment for that. So likewise, we can come up with some strategies which can minimize the occurrence of these adverse drug reactions. So in order to get that information, first we need to report and see the incidence. So out of 100 patients taking that medication, how many are showing that nausea and vomiting? Yes, it is known, but you should have some incidence. How many patients are having that problem out of 100 patients? So in order to collect all this, still if it is a known reaction or minor or non-serious, it is recommended to report it so that the analysis can be done at the higher levels.
Thank you, sir. Thank you for answering all the questions. And it was a very informative session, sir. Thank you so much.
Thank you.
So next, our next speaker for today's session is Dr. Sri Harsha Chalasani, Faculty, Department of Pharmacy Practice, JSS College of Pharmacy, JSS Academy of Higher Education and Research, Mysuru, and he's also the clinical trial coordinator in JSS Medical College and Hospital. We welcome you for this session, sir. So now I request Dr. Sri Chalasani to deliver the talk. Over to you, sir.
Thank you, ma'am. Ma'am, could you please confirm that I'm audible enough?
Yes, sir, you're audible, sir.
Thank you, ma'am. Can you all see my slides, ma'am?
Yes, sir.
Yeah. Thank you, everyone. And I thank everyone for giving me this opportunity to deliver the guest lecture on pharmacovigilance in the special populations. So to begin with, there is no conflict of interest. And for this presentation, I have utilized the material that is available at the India CDSCO, USA's FDA, and European Union's EMA. So to begin with, the previous speakers have spoke about how the pharmacovigilance has been defined. Just to have a quick understanding, it is the science and the activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problems. Now, this is a nutshell. We can say that it is the study of the safety of the marketed drugs under the practical conditions of the clinical use in the large communities, which is otherwise not possible during the clinical trials. When we are conducting a clinical trial, we have a lot of restrictions in the inclusion and the exclusion criteria, and we try to simulate as minimal noises as possible. Now, when this is happening and the molecule enters into the market, where by and large, the people can use these medicines under different permutations and combination, the way the safety concerns raises is quite understanding and quite underestimated either. So the pharmacovigilance, whatever we are doing it needs to be extended. The median number of the patients who are contributing to the data, that is the number remains too small to detect the uncommon or very rare adverse drug reactions or adverse drug events when we are carrying out a clinical trial. Even if they are serious, we might not be able to identify them, because in a global clinical trial, if you take the phase 3 study, we may hardly accommodate at approximately 10,000 of patients. But the global population may be something different, and we cannot generalize the findings to the general public. And the kinds of the patients, you see, those who are on the polypharmacy or having the multiple comorbidities and the special populations, vulnerable populations, are often excluded from the clinical trial to receive these medications during the clinical trial phases for various reasons, which we are going to look at it. Now, once the molecule enters into the market, we cannot restrict the usage of these medications with these special populations. And hence, the pharmacovigilance needs to be extended to these special populations, and it involves an array of complexities while you are determining the adverse drug reactions with them. But then, in India, once again, the healthcare professionals are too burdened to detect and report the adverse drug reactions effectively. An article that has been published in the Lancet 2014 says that for every 1,000 population, India is only housing 0.7 doctors. Now imagine they have to sit with the patient and they have to come down, do the causality assessment and come up with the right drug, that is affecting the patient's condition. Yes, so that could be really an arduous task. And for more so, the drugs are often withdrawn from the market for what they might have caused a very, very, very rare adverse drug reaction, and that has never been detected in their pre-licensing studies. I hope you all remember the pioglitazone study. Now, when we are coming about the special populations, today we include the pregnant women, children, and the elderly. So these special populations, they require the additional consideration with regards to the clinical research, because that is where the patient safety studies also take part. And the special population provides the enhanced awareness of their vulnerabilities. And once we can address them, the risk management becomes even more smoother and seamless. Now, when we are talking about the pregnant woman in India, the negative indicators that are associated with the pregnancy in India have been declined steadily. And then there have been a very positive pregnancies in India. We have seen that there is at least, the 90% of the pregnant women take one medication, and more than half of them they take at least one medication during their first trimester. So when it is happening, the pregnancy alters the drug absorption greatly due to the many factors. If you look at it, you see the way they experience the nausea and vomiting, it affects their food habits. The changes in their gastric volume and the pH associated with the acids, so once again, it impacts on their digestion, and often it can present as a condition for the constipation. Ultimately, when you look at it, all these factors, they inhibit or they alter or they impact the first primary pharmacokinetic parameter, that is absorption. Now, because of that, the consecutive states such as the metabolism and the transportation expressions, all of these would be affected greatly. Now, because of that, one cannot anticipate how this drug is being absorbed and how this drug is being, expressed in the pregnant woman. Additional factors sometimes also may, alter the pharmacokinetics, will indeed alter the pharmacodynamics, especially when you are talking the medications and the dosing in terms of the weight and in association with their body compositions. Pregnancies with the multiple children, multiple babies, such as the twins and the triplets, they can often alter the blood volumes. Thus, the volume of distribution is often disturbed. And thus, it ultimately boils down to one thing, that is the abrupt change in the use of the way the molecule behaves in the body. Now, this is only to be worsened by the concomitant usage of the alcohol and the tobacco when they are pregnant. Now, of course, this might not be the case with the most of them, but even if one person is going through any of these things, it will definitely impact not just the mother, but also the unborn baby. And further, the kidneys and the liver are majorly affected during the pregnancy. There is an increased intravascular volume, and that can lead to the 50% increase in the glomerular filtration rate. Now, as this happens, you see, these changes can sometimes, they prolong beyond the postpartum period. Now, because of this, you see, we cannot anticipate how this drug may be behaving in the body. Once the pharmacokinetic parameters are affected, then obviously the pharmacodynamic parameters are also affected. However, there is a conflicting in the evidence that is surrounding in the changes in the hepatic metabolism. Sometimes it is very, very unpredictable how one would react. In certain population, in certain women, there is an increase or there is a decrease in the hepatic metabolism during their pregnancies. and for some, it stayed unchanged. So this requires an additional monitoring in these women who are unchanged, who are experiencing the unchanged metabolic parameters. Now, we all know that how the thalidomide exposure to the fetus in the first trimester can cause severe impairment. And since that day, in the 1960s, we have come a long way to understand the safety use of the medications in the pregnancy. Specifically speaking, the use of non-steroidal anti-inflammatory drugs during the first trimester can increase the risk of the miscarriage and the malformation. While when they are used in the third trimester, it has the risk of premature closure of the ductus areatus. Now, all these conditions, how do we understand, and how large by large, how can we look into it? So that is where the pharmacovigilance has to be. And further, the drug interactions and the interaction between the drug and the development of each of the fetus during each trimester must be considered very, very significantly when we are determining the appropriate dosing for the pregnancy. Now, when you are looking at the pregnant woman as a research participant, it is understood that these research participants, pregnant women generally, they are willing to participate as long as it is a non-invasive research. This is a finding from the Western countries. However, in India, we have a very limited understanding about that. Now, any research that imposes the risk must take into account both the mother and the unborn as well. However, as this population is very, very important for us to, because there is an unborn baby, we should also account for the unprecedented adverse events that may occur. So whenever a study is being conducted, it is very important that we address these concerns adequately before we do the dosing. And the clinical research in the pregnant participants usually generates complex ethical issues and that requires a special protection, not just for the mother, but for the unborn baby. And this protection sometimes may extend to the lifetime of the baby. So considering all these complex ethical issues, this is one reason why the pregnant women are least taken into the clinical trials. Now, when you are looking at the regulatory aspects, in the 1994, the Office of the Women's Health was established by the US Congress in response to the zolpidol dosage. They thought that the men and the women would react same to the zolpidol. But then they realized that the woman would require twice the dose that the men might require, and thus the Office of the Women's Health was created. Now, one of the primary mandates of this office is to look how the gender differences, they are going to affect the medical product safety and its efficacy. And also, it tried to track the rate of the woman participating in the clinical trials, and it tried to educate on the effects of the unborn. Further, they have made sure that the pregnancy exposure registries are made available. These pregnancy exposure registries are the prospective observational studies and that actively collects the information on any medical product exposure during pre and post pregnancy, and they will counter it with the associated pregnancy outcomes. The US FDA hosts a list of pregnancy exposure registries, and these will be the hub for the information to understand how these drugs and the vaccines are affecting in the pregnant woman. And thus, we can have at least a core understanding whether this drug should be taken or it should be avoided. And I'm sure that, we already have the US FDA pregnancy class. But this is not, it keeps getting updated as and when we get the newer information on the safety use of these medications. Pregnancy exposure registries, you see, they differ from the post-marketing surveillance techniques. And one such I can tell that is about the birth defect registries and the spontaneous reporting of the adverse drug reaction. So we know that spontaneous reporting of the adverse drug reactions or the spontaneous reporting is the backbone for the pharmacovigilance activities. So these registries, despite you have it, if we cannot give the quality information into these registries and we cannot detect the quality outputs from these registries, then whatever the efforts that we are doing, they might not be substantiated in the further patient safeties. The other surveillance methods, that we may use are the retrospective studies. Whenever there is an adverse outcome, we will try to understand, or we will try to go back and see what was the mother is exposed with. But then it has its own share of biases. But then before we speak about the negative parts of the pregnancy exposure registries, what it can do is it can provide the margins of the reassurance regarding the lack of risk when a precise measure is impossible. Sometimes the situations may occur so that, you may have to, it maybe sounds so typically possible when we are talking about the risk and benefit analysis. But when it comes to the pragmatic decision making, it might not be the condition. So if such a condition to arise, these registers can give us a great deal of understanding. Okay, these many mothers have used this particular medication at this particular trimester and they were not affected by the child. However, most of the teratogens, they may follow all or none phenomena. Now, and another thing that we can also do with these pregnancy exposure registries are, we can monitor for the suspected risks raised by the preclinical studies and pre-marketing clinical studies. And at times, we can extend it to the post-marketing cases as well. I hope you all remember about the diethyl stilbestrol disaster. And then, because of this kind of registries, we can go back and identify the risk factors that can predict or that can cause in the pregnancy. Especially, we can boil down to the doses, times of the exposure, and the maternal characteristics. And thus, we can come with the predictors of such untoward outcomes during the pregnancy. And importantly, these registers will help us in creating the hypothesis. And once the hypothesis has been created, we can use the pharmacovigilance methods to either to strengthen the signal or we can rebut whatever has been generated. Hence, this plays an important, this is perhaps one of the best ways that we can have and which is easy to do it. However, the challenges are, as I told you, that the spontaneous reporting determines the quality of the whatsoever it is being reported. Now, it is not possible for every single physician to actively follow or track a pregnant woman right from the beginning till the end, and to make sure that they are receiving or whatever is happening with the patients. In India or in any country that is blessed with the traditional medications, you see we have multiple issues involving in the concurrent use of these traditional medications as well. Now, we have a very limited understanding about how these drugs interact in the conditions. When you give different kinds of medical, different types of the medicines to the patient, different systems of the medicines to the patient, all these conditions, you see, they may become a little bit of a difficulty for us to attribute a right drug for whatsoever the reaction has occurred. Now, another thing, if the lack of data, quality data, could always lead to the lack of a denominator for us. The moment we have to compromise on the denominator, we may also have to compromise on, generalizing how much percentage of the population or what would be the incidence of the population or what prevalence of this population is affected by this. Further, we cannot have any control to make sure that or to strengthen the signal in the pregnant woman. And moreover, the recall bias is associated with the retrospective reporting. And apart from all these things, there are barriers for the reporting. As such, we are facing a condition, we are facing a situation where the underreporting has become a normal norm. Because maybe whatsoever the reasons could be, because of the stress, because of the manpower, because of the understanding, or because of the complications involved in the reporting and understanding the causality assessment and making sure we identify the right drug and poor case documentation, all of them, they lead to the barriers under reporting. And these barriers could potentially compromise the very purpose of having the pregnancy exposure registries. It could be easy to operate, and the system should take on its own, irrespective of whoever is operating within the system. And another thing that we can also see that the poor case documentation that proves to be a bigger challenge than the underreporting. Now, the documentation and the archival of such documentation, it always seems to be the crucial and the crux for the maintaining to understand the safety studies in these pregnant women. If we don't have the complete details of the medication usage during the whole of the pregnancy process, we are lost. The whole purpose maybe. Now, there have been certain programs in the well-established pharmacovigilance countries, such as the STEPS or SMART or iPledge, to ensure that the pregnant women are deterred from the OTC medication usage. And they are also told about the risks involved in certain medications where they have to take for whatsoever the conditions they are prescribed. So whenever it is not always possible that we can prevail the benefit over the risk, and sometimes we may have to take a close call, a close margin call between the benefit and the risk, and we may have to administer the medicine. It is also possible that whenever a known human teratogen has been administered, it is not that every mother is going to give delivery to a child that is affected with a malformed baby. So we have to more understand the genomic predisposition, how the mother is going to be affected or how the unborn is being affected with these kind of medications. Now, moving to the next one, the pediatrics. Pediatrics are often referred to the therapeutic orphans right from the 1968s, and the condition doesn't change much to the current day scenario as well. Now, one of the major reasons for that to happen is because of the metabolic differences when we compare it to the normal adults. Now, we do know that there is a decreased clearance in the neonates and the infants up to 2 months of the age, and there is a sudden surge in the metabolic capacity, when you compare it to an adult right from the 6 months of age to the 2 years. So in another study conducted by the Ginsberg, they said that the many drugs that are primarily excreted renally are cleared at a faster rate than the adults, and thus they are experiencing the therapeutic benefit for a smaller duration compared to that of an adult. Now, the relative size and the perfusion of the organs also impacts how these children, how the pediatrics are, how these pediatrics are affected by the use of these medications. Now, when you look at it, the phase 1 metabolism, which is a most important phase where it turns a molecule into the more polar, is only at the 30% when it is compared to the adults. However, it would rapidly increase and exceeds the adult rates by the age 3 for certain drugs. By the time they attain the puberty, the phase 1 metabolic capacity is trending towards the adult levels, and it maintains the proper levels by the end of their puberty phase. And then the phase 2 metabolism is having much more greater variation, and it is independent of the growth of their age, and it is majorly because of the substrates that are involved in the conjugation reactions of the phase 2 metabolism. Further, the differences in the total body weight and the body compositions, they will directly affect the absorption and the volume of distribution of the many compounds. When we look at it, I know how the serum albumin or alpha-1 acid glycoproteins and their proportionalities and the quantity they are existing in the system, they would definitely once again do a bit of an impact on the volume of distribution. Children also, they tend to have the less acidic stomachs, and resulting in the better absorption of the acidic drugs. And sometimes they may also experience the delayed gastric emptying, which impacts the kinetics of the absorption and further the distribution. And also sometimes, because of the lower levels of the serum albumin, they may have the little less levels of the protein-bound drug, and thus the free drug concentrations may be attained very shortly, and thus you may have to be very careful when you're administering the medications. And moreover, when a baby is born, their blood-brain barrier is not often fully functional. And this may lead, the higher lipid profile drugs. Higher lipid drugs, they can gain easy access into the central nervous system, and the children and the newborn babies specifically, they may have unintended adverse drug reactions associated with the central nervous system. Now, whenever you're taking into the children as the research population, there is a lack of research in the pediatric pharmacology. It is because of partly due to the ethical issues related to the conducting of these studies. And another part is the lack of the financial incentives for the industry must anything go wrong. So use of exploration, extrapolation or exploration of the data that has been already brought by the adult population may not be sufficient enough to extrapolate onto the children population or pediatric population. But whenever we are doing it, we have to understand the extent of the application of the extrapolation of the adult data, the modeling, the pharmacokinetic modelings that we have used, and the simulation conditions that we have done for the adult population. They had to be overlapped onto the pediatrics only when there is a specific study related question is available. One should not generalize the findings of the adult population just as to the pediatric population. Once we can do this, then we can overcome a lot of limitations that are associated with the extrapolation of the general pharmacokinetic data that we are deriving from the adult population. So another problem that we may have is the invasive studies. Unless otherwise we conduct the invasive studies, we cannot be so sure in profiling the pharmacokinetic parameters in the pediatric population. And then most often this data, that whatever we derive from the healthy adults, you see, it cannot be transferable between the children. Once again, in the children, the rate of growth, the rate of how they physically mature, it impacts greatly on the pharmacokinetic properties, and thus it also determines the pharmacodynamic outcomes, and thus making them once again a special complex population. The biggest problem that we may have, undertaking the pharmacokinetic studies, especially when you're talking in terms of the newborns and the infants, it is related to the blood collection. And another thing is, whenever we develop an assay method, to understand or to further sort the pharmacokinetic studies, once again, its applicability to a specific population is always questionable. And when you are looking at the regulatory aspects, you see the Code of Federal Regulation, it only said that you can include children only if there is no greater than the minimal risk is presented. And the further reading can be done under the CFR Code 50 under subclause 55. However, it does not define what is a risk for a child. A minimal risk of, venipuncture that you do for the adult population to draw the blood can sometimes be a greater than the minimal risk for a child, and the outcomes could be unforgivable. So this is another reason why the most of the clinical trials or the clinical research organizations, they hesitate to take the children into the clinical research. Further, the volume of the blood or the volume of the, the perfusion rate and other things, they greatly impact the pharmacokinetic parameters. And if at all, if a child is experiencing an adverse drug reaction, another biggest problem that what we have is, they do not, they may not be able to effectively communicate saying that what they are suffering from. And most of the times we know that our physicians, our pediatricians, they use the off-label medications or unlicensed medications in these populations, such as sildenafil citrate for the pulmonary hypertension, bosentan. These are the classical examples that we use in the pediatric use. Now, whenever we are using these medications without approval from the regulatory agencies, there is an inherent fear coupled with this. What if something goes wrong? And what will happen post going wrong? So when you look at these things, you see the rate of reporting of the adverse drug reactions may not be very encouraging. And once we don't have an encouraging reporting pattern, we cannot generalize the findings of these particular pediatric population across the larger population. And another thing is the drugs that may be excreted, through the breast milk, we have a very, very limited amount of the data. It may be because of the poorly documentation or the challenges involved in identifying to what extent the drug has been excreted and how the drug has impacted the mother, the child. So all these are the certain complications that may prevent us from having the quality reports. Further, delayed adverse drug reactions, which have the longer latencies and that may have the impact on the effects of the growth, cognition, are less recognized in the pediatric population. Further, I would like to make a mention that they have come up with a color coding mechanism to identify the pleasantness of the drug consumption. Once a child has taken a medication, the doctor would present a series of array of colors where you see the yellow or the lighter colors are associated with the pleasant feeling, and the darker colors are associated with the unpleasant feelings post vaccination or post medication. And when they bring that to the child, and the child can subjectively select which color they are feeling, but then that would give us an understanding what the child is feeling after receiving the medication. But then the problem is these mechanisms are highly subjective, and they cannot give us a clear understanding, and they cannot give us a clear depth of to what extent these drugs are affecting them. So these are the other challenges we are having with the pediatric population. When you look at the geriatric population, the rate is steadily increasing, and from 8% in 2015, we are going to have 19% of them by 2050. So as they continue to progress, one of the biggest challenges that the geriatric population may face is receiving the multiple medications for their declining physiological functions. So when we are talking about the declining physiological functions, the polypharmacy poses a greater challenge to the achieving the appropriate therapeutic outcomes. Further, because of the altered pharmacokinetics and the declining organ functions and the comorbid conditions, the drug-drug interactions are often, they can precipitate into the adverse drug interactions. And more so likely, they are very hard to predict. Adverse drug events usually occur into the 15% of the elderly population, and only understanding that there is a great deal of underreporting. So whenever you look at the age-related changes, you see that physiologically they are badly affected. The gastric rate, right from the absorption, distribution, metabolism, and the elimination, and the amount and the extent of the blood flow to the organs, the perfusion rates, and the disturbances between the lean body mass and the fat body content, or adipose tissue extensions, and the capacity of the liver to continue the metabolism. All these things, they affect the overall pharmacokinetic parameters of these drugs. So what happens when they get affected with these pharmacokinetic parameters? Once a pharmacokinetic parameter is affected, there are likely two chances to occur. One, then it can lead to a therapeutic failure. Second, it can lead to an adverse drug interaction with the other molecules. And ultimately, it leads, the patient ending up deprived of therapeutic benefit. And in the pharmacovigilance, a therapeutic failure is also considered as an adverse drug reaction, and the pharmacovigilance has to take a call on. Now, when you are looking at the elderly as a research participants, the elderly patients with the reduced organ functions, they are often excluded from the clinical trial because of the stringent inclusion and exclusion criteria, and thus it makes it difficult for the clinicians to come down to the right amount of the doses that you have to administer, and how they would interact within the body, human body of an elderly patient. And another thing is having the comorbid conditions, and it just adds one more layer of the complexities to understand the journey of the medication within the physiological system. Ultimately, it would only complicate the situation in understanding how safe or how efficacious is this drug. Certain adverse drug reactions, it might require, I would rather say, it would require a clinical assistance in identifying them. Any chemotherapeutic agent and thrombocytopenia, they can only surface when you do the additional lab monitoring parameters. But it is not, also we know that the financial constraints of conducting these extra studies or extra lab parameters, in order to identify the adverse drug reactions could be very, very painful or a burden to certain populations in the elderly. So when you look at it, neither you can have them in the clinical trials, nor you can have them, in the post-marketing, because of the varied conditions such as the deprived of the declinity in the cognitive function, or the support system they enjoy, or the medications they receive, and their capability of expressing what they are going through, and often mistaking these medications or adverse drug reactions with the progress of the disease proves another layer challenges for reporting the adverse drug reactions. Usually, US FDA recommends that we should include elderly population who are 65 years and older. And also we should make sure that, there are studies, this is as per the US Government Accountability Office statement, at least 25% of the clinical trials, they should have the sufficiency of the elderly population. But it is not always possible. And another thing is, when we are determining an adverse drug event, itself could be a difficult thing during the clinical trials. Now, when it precipitates as an adverse drug reactions, they become much more difficult when it comes to the elderly population. The particular challenge is ADRs are commonly present as a symptoms, that are already prevalent in the elder population. Disease spells, falls, confusion. So there is a great deal of confusion for even for the healthcare professionals, whether it is an adverse drug event or an adverse drug reaction. The moment there is this confusion, it requires a greater extent of commitment to understand on what to report and how to report. And thus, another condition, the cascading prescribing. For one drug may cause one adverse drug reaction, and that adverse drug reaction may require another drug, and that another drug may again precipitate another adverse drug reaction or adverse drug interaction, and thus it continues to pillage the patient's overall mental health as well as the physiological. Now, when you look at all these challenges, you see, we have a greater deal that the pharmacovigilance has to put into these special populations. The mechanisms must be developed in such a way that, you see, the safe and efficacious use of the medications in the special population is always the first priority. But sometimes when we don't have the adequate conditions, where we can simulate the risk and take a call because of the presenting conditions, we have to be prepared also to stand to testify them in the regulatory agency bodies. One last thing before I conclude, when we are trying to look at the adult population data that we have received in the clinical trials, we should extrapolate them as we are going back from the elder pediatrics to the neonates. Meaning, whatever the data that you get, you may extrapolate them from 18th year to 17 to 16 to 15, and thus you can go down to the second 2 years of the age. But when you're talking about the older patients, you have to extrapolate this information 60s to 70s, 70s to 80, and 80s to further so on. And thus, we can have at least a basic understanding, if not, a clear picture how this drug is going to behave. Despite taking all the measures and all the cares, there could be a greater potential for an adverse drug reaction and an adverse drug event to occur. And it also demands the healthcare professionals are very, very strong enough in identifying, which is because of the disease progression and which could be because of the medication as such. And the registry study seems to be most effective and least, expensive that one can follow it. And these longitudinal studies will give us a great deal of understanding how these medications act or how these medications can, precipitate the changes in the later generations of the medications. One should remember the Faringham's heart study that has been conducted over 6 generations, and it is still continuing in understanding how the hypertension, how does this happen and who are more vulnerable to that. Ultimately, maybe it is my own presumption that the future could be of the pharmacogenomics and the precision medicine. Only with that one can answer how the safety profiling of these medications can be done without hurting a large, vast variety of the population. In the clinical trials, we have a stringent simulations where we exclude a great deal of the population to make sure that there is not so much of a burden that is happening. But when it comes to the market, we cannot run those strict simulations. We have to remember the permutation and combinations are infinite, and often we have to understand that the drug can often go wrong as it can go wrong. Again, not to forget about the cultural, cultural impacts on these medications, such as on the diet and on the fasting profiles and other things. When we look into all these things, you see, the pharmacovigilance in the special population is a challenging one, but definitely an enthralling one. So one who takes it up, I'm sure they would leave a permanent mark in the patient safety in the special populations. With this, I conclude my presentation. Thank you.
Thank you so much, sir. Thank you for your valuable information regarding the pharmacovigilance in various populations, sir. And we could hear about each and every population, even their challenges as well as limitations, sir. So it was very informative, and thank you so much for sparing your valuable time, sir. In your busy schedules, you have come and you have given a good presentation, sir. Thank you so much, sir.
Thank you.
So we can move on to our next speaker. Thank you, sir. So our next speaker is Dr. M. Ramesh. Dr. M. Ramesh, Professor and Head Department of Pharmacy Practice, JSS College of Pharmacy, JSS Academy of Higher Education and Research, Mysuru, and he's also heading the Clinical Pharmacy Department at JSS Medical College and Hospital, Mysuru. He has been graduated from JSS College of Pharmacy, UT in 1990 with completion of MPharm in Pharmacology, and from the same institute in 1992. And he has been awarded with doctorate in pharmaceutical sciences in April 2006 by Rajiv Gandhi University of Health Sciences, Karnataka, Bengaluru. He has also completed his postgraduate diploma in clinical pharmacy from the Society of Hospital Pharmacists of Australia, South Australia in 1998. He has received 10 months training in area of clinical pharmacy practice at Repatriation General Hospital, Adelaide, Australia, and was awarded Very Dunlop Fellow in clinical pharmacy from the same hospital. So he's pioneered in assisting Khartoum of Medical Sciences, Khartoum, Sudan, in establishing clinical pharmacy teaching as well as practice at ACMS and Bashir Hospital, Khartoum, Sudan. Also, he imparted World Health Organization sponsored clinical pharmacy training program to pharmacy teachers from 6 different universities in Sri Lanka. He has been coordinated for more than 50 clinical trials of various phases in different therapeutic areas, including global trials. Also, he presented several scientific research papers in various national and international conferences. He has successfully guided 71 postgraduate pharmacy practice and PharmD students. He has successfully guided 4 PhD students as well as currently guiding 7 PhD candidates. He held and he has been holding several posts at various capacities in different academic as well as professional bodies, and he's a member of various professional bodies like IPA, APTI, IHPA, ISPE, and ISPA. He has visited several countries for academic purposes, including Australia, Singapore, Sudan, Sri Lanka, Dubai, Thailand, Hong Kong, Turkey, Denmark, Austria, Malaysia, China, and Canada. Now I request Dr. M. Ramesh to deliver the talk. Over to you, sir. So you are not audible, sir. So there's a second option above the screen. So if you're clicking on to that, audio option will be on next to the video options. So please on your audio option. Yes, sir.
Yeah. Am I audible?
Yes, sir. You're audible.
Yeah. Okay. I think it's taking time, yeah. Is the screen visible?
Yes, sir. It's visible, sir.
Okay. You can see the full slide?
Yes, sir.
Okay. Yeah. Thank you, Dr. Rossi, for your kind introduction. Very good afternoon to you all. The topic that has been assigned to me in this webinar session is causality assessment of adverse drug reactions. For the next 30 minutes, I would be talking to you on why causality assessment is important and what are the different approaches that are available for assessing the causality. Then what are the data elements that are required for the causality assessment, and also the different methods by which we would be able to assess and assign the causality category of the reported event. As you all may be aware, in the clinical practice, the terms adverse drug event and adverse drug reactions are used as synonymous, but they are not synonymous. The term adverse drug event basically refers to the reported information upon which the causality has not been established. Once the causality has been established, then the adverse drug event will become an adverse drug reaction. Causality assessment is nothing but establishing the likelihood relationship between the suspected drug and the reaction. It is usually done by using the well-structured and standardized questionnaires, scales, or the algorithms. In assessing the causalities, there are two issues that need to be considered. Firstly, is it possible that the drug can cause the adverse drug reactions? In other words, whether the reported event is a known and it is well documented in in the published literatures? If yes, if it is well documented in the published literatures, then as the drug actually caused the event in a given patient. That means you need to check whether the particular drug has caused a particular reaction in that particular patient. For example, if you take a report of paracetamol-induced fixed drug eruptions, firstly, you need to check whether is it a known reaction. If it is a known reaction, whether it has been well documented in the published literatures. If it is well documented, then you have to look at whether that paracetamol has caused that particular reaction, that is a fixed drug eruptions in that given patient from whom the report was made. So these are the two issues that need to be considered. Now the question is, why assess causality? Causality assessment is important for several reasons. Firstly, it is legally required by the various health authorities to report and serious adverse events, be it US FDA or European Medicine Agencies or Indian CDSCO or any other regulatories. If you want to report a serious adverse event, the causality assessment should have been done. Secondly, it is important to incorporate the reports into signal detection review process. For example, if you want to consider the report in the process of signal detection review, then the causality should have been assessed. Similarly, to consider the report in the risk-benefit analysis, the causality should have been done. And also, if you want to take the report to make any decisions on listing of the adverse events in the labeling, whether it is in the package insert or it may be in the summary of the product characteristics, the causality should have been done. So without doing a causality assessment, we cannot consider the reported event to any of these processes, including the signal detection or the risk-benefit analysis or taking the decision on listing of the adverse events in the labeling. And another important reason why causality assessment is important is it is important to warn patients, investigators, and other healthcare professionals. Because unless otherwise, the causality assessment is done and the association between the reported event and the drug is proven behind the doubt, one should not alert the healthcare professionals or the patients or the investigators. If you want to alert them that so-and-so drug can cause so-and-so reactions, the causality assessment is very, very important. There are different approaches to causality assessment. The first approach is global introspection. Otherwise, it is called as clinical judgment. As the term implies, in the global introspection, here an individual or the group of individuals who are experts in the area of pharmacovigilance would assess and assign the causality of the reported event based on their clinical knowledge, their expertise, and also the amount of information which they have with them on the reported event. The major drawback with this global introspection is there are greater chances of the inter and intra-individual variability. See, inter-individual variability means, for example, if you give a data on the reported event to two different clinicians, one may say it is probable, another may say it is possible. This is mainly because it depends upon to what extent the individual has perceived that reactions and about his expertise and his understanding, okay, that decides what would be the causality. Similarly, there are chances of the intra-individual variability. Even if you give the same reported event and ask the same individuals to do the causality assessment at different points, one point he may say it is probable and another point he may say it is possible. Because it depends upon at the time of assessment, what is the mental status and to what extent you would recollect the informations that is available with him and to what extent he would apply his knowledge in analyzing the causality. So that means even the same person analyzing at different point of time, one time he may say the possible and another time he may say that it is probable. So that type of intra-individual variability is also possible in the global introspection approach. But this inter and intra-individual variability can greatly be minimized in the formal algorithm approach. In the formal algorithm, we would be using a well-structured and standardized scales or the questionnaires, okay, or the other standard criteria. Since we are using a well-structured and standardized questionnaire, scales, or the standards, it can improve the scientific validity. If not to a greater extent, at least to some extent, there will be a scientific validity, because we are using, even if it is a questionnaire or criteria or whatever which are using to assess the causality, it is the validated one. So that certainly there will be the greater amount of the scientific validity in that. But the drawback is it is a time consuming. It is not that time consuming, but when compared to the global introspection, it is relatively time consuming. Okay, for example, if you say global introspection, one can do this in 5 minutes time, okay, and it may take another, another 5 minutes extra, maybe 10 minutes of time is required in case of formal algorithms. I'm talking about 10 minutes of time. It is only for the assessment, stating that you have all the data with you. You're just simply only assessing it. But otherwise, it may take an half an hour to 1 hour to assess a particular the event, especially when you have to gather the data and then do the assessment. Okay, and what I'm talking about 5 to 10 minutes is only to you have all the data with you and just only to apply the data and see by using the standard questionnaires or algorithm and then assess the causality. And the third approach is it is a probabilistic Bayesian analysis and other statistical methods. This method generally requires more data than that is available. And it is not practically applicable because it needs more data, and also it needs minute data with quantification, with the quantifications. So sometimes, it takes maybe a week time to assess a single case report. So that means it is practically not possible. Therefore, out of all these three different approaches, the most commonly applied approach in the clinical practice is it is a formal algorithm wherein we would be using a well-structured and standardized questionnaires or the scales. The assessment of reported areas involves two stages. The first stage is assessment of individual case safety reports. We call it as an ICSR, that is individual case safety reports. In individual case safety reports assessment, when an event is reported, one would gather all the required informations to assess the causality, and then he would apply all those data, okay, and his expertise to see what is the likelihood relationship between the reported event and the reactions. So this would be done soon after the event is reported, and it will be done when you have an ADR of small numbers, and you will be doing there and then an event is reported and immediately you can do the causality assessment, provided that you have all the data to assess the causality. Whereas the second stage, it is the interpretation of the aggregated data. When I say the interpretation of aggregated data means it is the aggregated data of similar reports. For example, if you say amoxicillin induced diarrhea, you should have multiple reports of the amoxicillin induced diarrhea. It is not that you have few reports amoxicillin induced diarrhea and few reports you have amoxicillin induced rash. That kind of reports you cannot have. You should have the drug should be the same and the event should be the same. That's what you say, that interpretation of the aggregated data. And this kind of interpretation of aggregated data is done, especially during is done to generate the signal. That is to identify the new reactions. In identifying the signal signals or the new reactions, usually they say that you need to have multiple reports. That means, some of the literature says you need minimum of 9 reports to generate the signal. But at the same time, some of the literature says you need 6 reports or 3 reports. And sometimes even, you can also do the signal detection even with a single report, especially when the reported event is a very, very, very rare report, because you would not be able to get, multiple reports because it is a very, 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 for example, if it is in a 1 in, 50,000 or one in the incidence is 1 in 50,000 or 1 in 1 lakh. So such kind of reports you would not be having in multiple numbers. So even if that kind of rare reactions, you would be able to do the causality assessment or the signal detection with one single report itself. Okay, but otherwise in general you need a very large number of ADR reports of the similar reports. So in assessing the causality, we need to have certain data elements with us, because there are certain important data elements which we should have it before we start the assessment of causalities. Now let us see what are those data elements. The first data element is the complete knowledge of clinical event. When I say that complete knowledge of clinical event, means it is a complete description of the event. It is not enough just to know that what is the nature of the event, but you should have a complete description of the event. For example, when an headache is reported, most of the time what happened is they say that, they just mention that event as a headache. Okay, that may not be sufficient. So when they report it, they have to give a complete description, like for example, whether the headache is one-sided headache, or it is a double-sided headache, or it is a dull aching pain, or it is a very intense pain, whether it is an intermittent pain or it is a continuous pain, whether the patient gets the relief from the pain when he takes the rest, or when it worsens when he is involved in some physical activities. So something like that kind of details need to be given. That's what we call it as a complete description of the event. So one should have a complete knowledge of the clinical event. Okay, it is not sufficient just to know what is the event. The second data element is the patient's medical and medication history. When you say the patient's medical history, means one should know, you have to gather the information on what are the presenting complaints, what is the provisional diagnosis, what is the past medical history, what is the past surgical history, and what is allergic status. So likewise, you need to gather all the patient's medical conditions before you do the assessment. Similarly, one needs to gather the complete medication history of the patients. When you say the medication history, means you need to gather the information pertaining to current medications, then medications on admissions, OTC medications, and also try to understand whether the patient was taking any other system of medicines, be it Unani or Ayurvedic, Siddha, whatever it is. So you need to gather all this kind of the details of all these kind of medications, including current and past medications. When we say that the medication details, means it is not that just knowing the name of the drug. Okay, you have to gather a complete dosing regimen. Okay, complete details of the each and every medications which the patient was taking. For example, what is the drug, what is the brand, and what is the dosage form, and what is the strength, and what is the route of administration, and what is the formulation that is being used, and what is the duration of drug use. So every details need to be gathered. It is not only that these details need to be gathered only for the suspected drug, but also the other drugs which the patient was taking at the time of experiencing the ADR. And it is not only for the current medications, but also you need to gather this information for OTC medications if the patient is taking, if any, and similarly for the medications on admissions, etc., etc. And the third data element is the temporal time relationship. It is a very, very important data element. That is a temporal time relationship. Temporal time relationship means it is the time gap between the drug administration and the occurrence of the event. That means when did the patient take the medications and when did the reaction occurs. What is the time gap? Whether that time gap is plausible to cause the reactions. That is very, very important, because in all the time, it doesn't mean that the patient develops a reaction soon after taking the medication. You cannot associate with the drug. Okay, for example, the same example I can take. Paracetamol-induced fixed drug eruptions. You say when the paracetamol is given by oral route. Okay, patient has taken the tablet paracetamol, and after 5 minutes, if he would complaints of rashes or the fixed drug eruptions, whatever it is, and it doesn't mean that just because the reaction appears after taking the paracetamol, okay, and paracetamol is involved in causing that reactions, not necessarily, because we know that here the reaction has appeared only 5 minutes after taking the tablet paracetamols. Okay, so when a patient takes the medications orally, we all know that it would take at least minimum of 15 to 20 minutes of time to elicit its response, because in the oral dosage form, it has to undergo disintegration, dissolution, and then it has to reach the blood circulation and the receptor site to elicit any response. But at the same time, if the paracetamol is given by IV route, okay, and the patient develops such reactions soon after that, then there is always possible, because in IV you are directly placing the drug in the veins, and obviously it can cause the reactions immediately. So that means you need to understand exactly what is the time gap between the drug administrations and the occurrence of the reactions, and whether is there any a plausible time relationship between these two. That is very, very important. And another important data that is required is, or that is on the previous reports on the reported reactions. You need to understand whether the reported event is a well-known reactions, whether it is previously well documented or not. If it is well documented, then what is the incidence of that particular reported event? So because all this information which would help in the causality assessment, especially in strengthening the involvement of causing this particular drug in that particular reaction. Similarly, the other data that are required for the causality assessment is, apart from those 4 data, possible alternative causes. When you say that possible alternative causes, it could be a drug-related causes or it could be a disease-related causes. So that means that is the reason why we need to gather a complete information about the patient medical conditions and as well as the medications. Because, for example, when the patient is taking the 4 medications and the patient develop an ADR, and the clinicians may suspect to any one drug, and he may make a report to any one drug. So that means if you want to attribute, okay, if the patient is taking the drug A, B, C, D, 4 drugs, okay, and it is attributed to drug A, then if you want to attribute to drug A, then during the causality assessment, you have to rule out the involvement of drug B causing that particular reaction. Similarly, the drug C and drug D causing that particular reactions. So that means you need to rule out all other possible causes. It is not only related to the drugs which the patient was taking at the time of experiencing the ADR, but also the other disease conditions. Because sometimes the new symptoms which the patient develop may be the disease process itself. It may be a part of the disease process, or it may be because of the comorbid conditions which the patient was having. Okay, comorbid conditions or the intercurrent diseases or anything. Okay, so it is very important to rule out all those possible causes. So that means you need to gather all that other possible causes and then assess and rule out all those factors, okay, to attribute to the particular drug to the particular reactions. Other important data is the D-challenge and the re-challenge. One should know what is the outcome of D-challenge. The term D-challenge means it is a cessation of the suspected drug. So here it is not only enough to know, okay, whether the drug is cessated or not, the suspected drug is cessated or not, but you should know what is the outcome of that. What happened after cessating the drug? Whether the patient has recovered from the reaction or the reaction continue to exist in the same intensity, or it has worsened further. That is very, very important. If the patient has recovered from the reaction after stopping the medication, then that is a suspected medication, then you would say that it is a positive D-challenge. If not, it is a negative D-challenge. Similarly, you also need to have a data pertaining to the re-challenge. That is the outcome of re-challenge. The term re-challenge means it is a re-administration of the suspected drug. When you say re-administration of the suspected drug, you need to administer the medication, the suspected medications, okay, in the similar manner how the drug was administered previously. For example, previously if the drug was administered in a oral dosage form in a tablet, the second time when you do checking for the re-challenge, once again you should use only the tablet. Okay, so in terms of all these dosing regimens, it should be ditto, like in terms of drug, in terms of dose, in terms of formulations, in terms of route of administrations, everything should be ditto how the patient had taken the medications prior to experiencing the reaction. That is very, very important. And another important point you need to understand here is re-challenge is possible only when there is a positive D-challenge. That means the suspected drug was cessated and the patient has recovered from the reaction. Only then you would be able to do the re-challenge. If the suspected drug is not stopped or it is stopped but the patient did not recover from the reaction, then the re-challenge is not possible because already the reaction exists in that patient. Okay, it is possible only when there is a positive de-challenge. And re-challenge we would not be doing in all cases, and because it is considered to be unethical in doing in all cases. That's why if you look at in clinical practice, about in 99% of the cases, okay, we would not be doing the re-challenge. Okay, this re-challenge will be done usually in conditions where it is medically warranted. Okay, it is medically warranted. That means there are no other medications to treat the particular medical conditions. Okay, and patient develops certain serious reactions to that. Then you would be, stopping all these medications, and then you see that, okay, whether that drug has caused that particular reactions or not. Okay, especially to give one example, I can say that ATT drugs. Okay, ATT drugs are highly hepatotoxic. Okay, the patient taking the ATT drugs develops an hepatotoxicity, then you would be stopping all these, medications and introduce one by one. Okay, to find out which ATT medication is responsible for causing that particular reaction, that is hepatotoxicity, so that you can stop administering that medications and continue with the other medications and go with the second line agents as well. Okay, so that is how you should be doing it. So similarly, re-challenge, when you do re-challenge and the same, if the patient develops a similar reaction, then you say it is a positive re-challenge. If the patient do not develop the similar reaction, then you say it is a negative re-challenge. Whenever you say that re-challenge, once again, the patient should develop the similar reaction, what the reaction he has developed previously. Okay, for example, initially he would have developed fixed drug eruptions, and later he would have developed the rash, then it is not a positive re-challenge, because first time, he has developed the fixed drug eruptions, okay, to a drug, but later when you reintroduced, okay, and he has developed not the fixed drug eruptions, but he has developed the skin rash, then it is also not considered to be the positive re-challenge. Only when he develops the fixed drug eruptions, that is a report which was made earlier, okay, only then you say it is a positive re-challenge. So those are some of the criteria or the data elements that is required for the causality assessment. So there are certain methods are available to assess the causality. Okay, there are several methods are available. I have just listed a few methods here. That is important methods. One is the WHO assessment scale. Another one is the Naranjo scale. Then European ABO system. Then you have Carson-Lasagna scale and Kramer scales. Then Bayesian neural network, Yale algorithms, and Spanish quantitative imputation system. Of all these methods, the most commonly widely used methods across the globe is a WHO assessment scale and the Naranjo scale. WHO assessment scales and the Naranjo scale. And to some extent, especially in European countries, some of the European countries, they use the European ABO system. Okay, but otherwise it is the WHO and the Naranjo scales. Now, among this WHO assessment scale and the Naranjo scale, okay, if you have to report an event to the National Pharmacovigilance Center across the globe, okay, because every country has their own national pharmacovigilance system. Okay, if you have to report the event to the National Pharmacovigilance System or to the regulatory authority, the causality assessment should have been done by using the WHO scale. They do not accept the causality assessment made using the Naranjo scales. But however, Naranjo scale is widely used, okay, amongst the researchers, okay, among the researchers. When they do this, the research, okay, they may use the Naranjo scale for the ease of use, okay, but otherwise it is ideally the WHO scale is recommended for the causality assessment, especially when you're reporting the event to the National Pharmacovigilance System or to the regulatory authorities. So since WHO assessment scale and Naranjo scales, these are the two scales which are widely used, we should be discussing more about these two scales. That means how we can assess the causality by using those two scales. Okay. When you are assessing the causality, okay, you would be ultimately end up with categorizing the causality of the reported event. What you need to understand here is the causality of the reported event varies with the scales or the methods which you adopt. For example, if you take WHO scale, if you are using the WHO scale for the causality assessment, you would be able to categorize the reaction into any one of the 6 categories, maybe a certain, probable, possible, unlikely, conditional, or unclassifiable. Similarly, if you take Naranjo scale, you would be able to assign to any one of the 4 categories, because Naranjo scale has only 4 categories, whereas the WHO scale has 6 categories. So in Naranjo scales, you would be able to categorize to any one of the 4 categories. Okay, namely, you say that definite, probable, possible, and unlikely. Similarly, as I said, some of the European countries, they use European ABO system. If you use the European ABO system, you would be able to categorize into any one of the 3 categories. For example, you say it is a probable, possible, and unclassifiable. That means in European ABO system, there is no certain category or the definite category, whereas this certain or definite category is there in the WHO and the Naranjo scale respectively. So that means if you are using the European ABO system, you would not be, even if you have all the data with you, okay, including the rechallenge, dechallenge and rechallenge information, and the rechallenge is positive, still you are able to assign only to the maximum of the probable category. Okay, but at the same time, if you use the WHO scale, you would be able to assign to the certain category. So that means the causality assessment basically varies, okay, depending upon what type of scale which you are using it. Okay. At the same time, the causality also can be determined by the documentation of the case report and as well as the evidence that are available. In other words, it all depends upon how much of information which you have and whether these informations are the factual one. Okay, that is very, very important. For example, even you are using a WHO scale to assess the causality, and you do not have any information on the rechallenge, because as I said, in most of the cases, in 99% of the time, they don't do the rechallenge. That means you will be lacking with the rechallenge information. So that means the maximum causality category that you can assign, okay, if provided that the dechallenge is positive, it is only the probable. It is only the probable. But at the same time, if you have a rechallenge information with you and that rechallenge is positive, then okay, in the same scale, you would be able to assign the causality category to the certain. Okay, so that means whether it is certain category or it is probable category, okay, it is once again depends upon the amount of information which you have, okay, and also the factual informations which you have with us prior to the assessment. So that means even the amount of data and the evidence which you have also can determine the causality of the reported ADR. As I said earlier, WHO scale and Naranjo scales are the 2 scales which are widely used. Okay, let us see how we can assess and assign the causality category by using these 2 scales. If you take the WHO scale, okay, as I said earlier, we would be able to categorize into any one of these 6 possible categories. Okay, one is the certain, other one is the probable, other one is the possible, then unlikely, conditional, or unassessable, or unclassifiable. These are the 6 possible categories for the causality category if you are using the WHO scale. Now let us see that, okay, when we can place the reported event, the causality of the reported event to a certain categories. If we had to place or categorize the causality of the reported event to a certain category, it should fulfill all these 5 criteria that are listed in this. Okay, so what are those 5 criteria it has to fulfill to be a certain category? Is one is the event with plausible time relationship to drug intake. That means temporal time relationship. Now we can go back to what we discussed about what are the data elements that are required for the causality assessment. Okay, this all refers to that only, these categories. So one is event with plausible time relationship to drug intake, means it is a temporal time relationship. That is the one. If it is that there is a temporal time relationship, then yes, there is an association. Then no other explanations. Either it could be disease or the drugs. That's what I said, under the data element, that we need to rule out all other possible causes. That means you are trying to rule out all the disease-related and drug-related possible causes. If there are no other explanations other than only the suspected drug, then third one is the event definitive. Event definitive means once again whether the reported event is previously a noun reaction or not. Okay, whether it is well documented or not. If it is a noun reaction and it is well documented, yes, there is a strong association. Okay, so that is the third point you need to consider. Then response to withdrawal is plausible. When you say response to withdrawal is possible, means it is nothing but positive dechallenge. That means you cessated the suspected drug and patient has recovered from the reactions. Okay, and that is the 4th criteria. And the 5th criteria is positive rechallenge. That means you have done the rechallenge and patient has developed the similar reactions to the similar drug. Okay, so when a reported event fulfills all these 5 criteria, then you would be placing it under certain categories, okay, as far as WHO scale is concerned, okay. And here, the key important point is it is a positive re-challenge. That means, unless otherwise the re-challenge is positive, you cannot place the causality into certain categories. It is a must to place a causality category to the certain category. That is a positive re-challenge is must. That's why if you look at, in most of the time in clinical practice, the maximum possible probability causality category is it is only the probable, because re-challenge is not done. That means you do not have a re-challenge information to assess the causality. Ultimately, you cannot categorize that into the certain category. That is the reason why in most of the instances, in most of the cases, in clinical practice, okay, the reported event would be categorized into either probable or the possible categories. So when do we categorize that causality into probable category? Okay, by when you use the WHO scale is, okay, when it fulfills all the first 4 criteria, but the 5th criteria, that is the re-challenge, okay, that information is not available, or re-challenge is done, but you do not know what is the outcome, or the re-challenge is done, you know the outcome, but you know that it is a negative re-challenge, not the positive re-challenge, then you would be simply categorizing to the probable. That means it will fulfill first 4 criteria. That means there is a temporal time relationship, no other explanations, okay, and there is a positive de-challenge, okay, but only thing is you do not have the information on the re-challenge, or if you have information and you know that it is a negative re-challenge, okay. But to place the causality category into probable category, once again, the key point or the key feature is it is a positive re-challenge. That means to place the causality category into probable, you need to, the de-challenge should be a positive, okay. Otherwise, you would not be able to categorize under this category. So to place the causality into the possible category, even if you do not have the de-challenge informations, or the de-challenge information is not available, or the de-challenge is negative, okay, but if there is a temporal time relationship, okay, but there is a temporal time relationship, you will be able to simply categorize under the possible. So the major feature to place the causality category under possible category is it is a temporal time relationship, okay. Even if you do not have re-challenge information, even if you do not have the de-challenge information, also not an issue, okay. Only thing is you have to ascertain, yes, there is a temporal time relationship between that, okay. Sometimes it could also be explained by the disease, it doesn't matter, okay, but major criteria is the temporal time relationship. If that is there, then you place it under possible categories. But when the temporal time relationship itself is doubtful, okay, when the temporal time relationship itself is doubtful, then you would be simply categorizing into the unlikely, okay. So that is what you do there. So sometimes even maybe a disease or other drugs are also give some possible explanations, or they might have involved in that causing that event, and you do not have a de-challenge information or the re-challenge information, okay, all this fine, okay, but temporal time relationship itself is doubtful, then simply you place it under unlikely. Then another classification under WHO scale is a conditional. Conditional means event is reported and you have some information with you, but that amount of information which you have with you is not sufficient to assess the causality categories. Okay, you may need more additional informations. So that means you are waiting for some additional informations. At that time, you can simply say that it is conditional. That means you are waiting for some more information. Once you get additional information, what is required for the causality assessment, then you can reassess, and then based on the amount of information and the evidence, then you would be able to categorize it into possible, probable, unlikely, or certain, whatever it is. Okay, so that is what the conditional category is all about. Then the final category is the unassessable or unclassifiable. Whereas unassessable and unclassifiable it is similar to that of conditional only. You have a reported event, but at the same time you have only very limited information. But the difference here is, you know that you would not be able to get the further additional data. But whereas in the previous one, conditional, you know that you would be able to get a previous data. You may wait for a day or two, gather the additional information, and you would be doing the causality assessment, redoing the causality assessment. But here, once you know that there is no, you have limited information, but at the same time, you will not be able to gather the further additional information, then you should simply classify that under the unclassifiable or unaccessible. Okay. For example, patient might have discharged against the medical advice. The event is reported. Okay. By the time you want to gather the all additional information, patient would have been discharged against the medical advice, or whatever the reason is. Okay. That means you don't have further information. You know that you are sure you will not be able to get that information. So classify under unclassifiable. So coming to the Naranjo scale, if you are using the Naranjo scale, you would be able to categorize it into any one of these 4 categories, namely definite, probable, possible, and unlikely. Now, Naranjo scale, okay, Naranjo scale has a set of, it is very simple and easy to use. That's why, most of the time in the, the academic researchers, they would use Naranjo scale rather than the WHO scales, okay. In the Naranjo scale, you have set of 10 questions. What you need to do is you need to answer to each and every questions of that. And depending upon your answer, okay, you would be, you would end up with scoring some points, okay. And the possible answers is yes, no, and do not know. So do not know means you do not have any information, okay, about that questions, okay. Then when you say do not know, when you say do not know, you do not have any information, then you have to be neutral. That means when your answer is do not know, you will be simply scoring 0. Okay, that's why if you look at do not know answer, the scoring is, okay, it is 0 for all the questions because you have to be neutral because you don't have information on that. But if your answer is yes or no, then you would be able to score some points. These points may be in the positive side or it may be in the negative side depending upon the question and depending upon your answers. Okay. Now, what are those 10 questions, okay, which you need to answer is, the first one is, are there previous conclusive reports on this reaction? That means that's what, whether it is a well-known reaction, whether it is well documented in the previously in the published literatures, that you have to say. If you say yes, then he would be scoring +1, no means 0. Then the second question is, did the adverse event appear after the suspected drug administered? That is what the temporal time relationship. If there is a temporal time relationship, you would score 2 points, otherwise you would score -1. And the third one is, did the adverse reaction improve when the drug was discontinued? That means it is what the outcome of D-challenge. That means you have D-challenge, that means you cessated the suspected drug, then what is the outcome of it? If the outcome is positive, then you would score +1, otherwise you would score 0. The fourth question is, did the adverse reaction reappear when the drug was re-administered? It is what the re-challenge is. It is all about re-challenge. If it is a positive re-challenge, okay, then you would score 2 points. If it is a negative re-challenge, you would score -1. Then the fifth question is, are there alternative causes? That's what I said, you need to rule out all other possible causes. If you are able to rule out all other possible causes, okay, then you say that it is +2, and then otherwise you would score -1. And the sixth question is, did the reaction reappear when the placebo was given? Okay, yes, you would score -1, no means you would score +1. But at the same time, if you look at in Indian settings, okay, this not only in Indian setting, most of the cases, okay, even worldwide, this placebo is not given to the patient to check whether the patient develops the reaction what he has developed to the reported drug. Okay, so most of the time, you would not be having any information. Then you simply say that do not know, and you would end up with scoring only the 0. Similarly, was the drug detected? And the next question is, was the drug detected in the blood? That means nothing but TDM, therapeutic drug monitoring. And you know that in most of the cases, it is not being done. Okay, and obviously he would end up with scoring a 0 for that. Okay, 0 for that. And the eighth question was the reaction more severe when the dose was increased or less severe when the dose was decreased? And this question is applicable only when the ADR is a type A reaction. It is not applicable for type B because we know that only type A reactions are the dose dependent, not the type B, because type B reaction are dose independent. Okay, so it is applicable for only such kind of reactions. And depending upon your answer, you will score. Yes means 1, otherwise it is 0. Then the ninth question is, did the patient have a similar reactions to the same or in any previous exposures? Okay, that means the allergic reaction, whether this patient has developed the same reactions in the previous situations. Okay, if yes, +1, otherwise it is 0. Then final question is, was the adverse event confirmed by any objective evidence? Okay, so it may not be possible to have an objective evidence every event, because some of the event may have an objective evidence and some of the event may not have an objective evidence. For example, if we take a reported event is in a rash, it is only the subjective evidence. There is no objective evidence. If it is an hepatitis, yes, you have both subjective evidence and as well as the objective evidence, because you can measure the liver enzymes. Okay, measurement of liver enzymes, if it get elevated, you say that, then you say it is an objective evidence. Okay, so it may not be possible in all cases. It depends upon the event that is reported, and you have to see the case by case and then see whether is there any objective evidence, and if objective evidence is there, whether objective evidence also present in that particular case. Okay, so depending upon that, you would be scoring either +1 or the 0. Okay, so these are some of the 10 questions, okay, in the Naranjo scale, which you would be answering, and depending upon your answer, you would end up with some scores. Okay, and at the end, after answering to all the 10 questions, you would be, you would simply sum up the score, and based on the total score, you would be able to categorize that. Okay, if the score is 9 or above 9, then you say it is the causality category is definite. If it is between 5 and 8, then it is probable. If it is between 1 and 4, it is possible. If it is 0 or less than 0, then you say it is unlikely. Less than 0 means it can go in minus also, because you know that some other questions, you would end up with scoring minus as well. So, okay, it may be in the score, maybe even minus also. So depending upon the total score, you would be able to classify the, categorize the reaction into any one of these 4 categories, okay, using the Naranjo scale. Okay, so this is how you would be able to assess the causality category by using the Naranjo scale. So now the question is, why do we use, as I said, there are 3 approaches. One of the approaches is algorithm. Now the question is, why do we use algorithm? I said in clinical practice, we use only the algorithms, not the global introspections or not the probable, the statistical approach. Okay, it is mainly the reason is, okay, it has certain advantages. One of the major advantage of this is, as I said earlier, it decreases the disagreement between 2 assessors. Okay, as I said, that is a major advantage. That means it greatly reduces the intra-individual variability. Okay, that is one of the major advantage. Secondly, it can classify the uncertainty. That means it has the ability to elicit the relationship between the reported event and the drug. Okay, that's what this. It can classify the uncertainty. Okay, that means relationship is, what is the relationship between the suspected drug and the reaction, whether it is probable or it is possible or it is unlikely, whatever it is. The third advantage is it has the ability to mark individual case safety reports. That means every event, the adverse event that is being reported, you would be able to assess for its causality and you would be able to categorize it. And the fourth advantage is it improves the scientific basis of assessment. As I said earlier, we use in algorithm, we use a well-structured, standardized and validated questionnaires or the scales. Okay, and obviously there will be scientific value to that. Okay, so there is some kind of scientific value to the causality assessment. Okay, so these are some of the advantages, why we are using these algorithms. Okay. When we say these are some of the advantages, it doesn't mean that it is not devoid of limitations. It has its own limitations. Okay. One of the major limitations is accurate quantitative measurement of likelihood relationship is not possible. That means you cannot do the quantitative measurement. What you are doing is, it is qualitative. It is in a more of relative terms. For example, you would categorize as like, probable. It is probable that the drug has caused that reaction, or it is possible that the drug has caused the reaction, or it is unlikely that the drug has caused the reaction. So it is a more of a relative term and it is in a qualitative manner. Okay, so quantitative measurement is not possible here. Okay, that is the one limitation. Second is it fails in distinguishing valid from invalid cases. That means it cannot differentiate whether the reported event is a valid report or it is an invalid report. Whether it is a valid report or invalid report, still you would be able to assess and assign the causality category, even for the invalid reports. Okay, so that means the data, you may not have a complete amount of complete data. At the same time, some of the data it is not a factual data. Still, if you assess that, you would be able to assess and assign the causality category. So that is another drawback with that. And then the third limitation is, it cannot prove the connection between a drug and the event. Whatever the assessment you do, it can just elicit the likelihood relationship between the suspected drug and the reaction, but at the same time, it cannot prove, the connection between the drug and the event. That means, in reality, we do not know. We do collect some data and assess and say this is what the causality category is. But in reality, whether the particular drug has caused that particular reaction in that particular patient, no one knows in reality what exactly happened. Okay, so that is another limitations of that. Then the fourth limitation is it fails in quantifying the contribution of a drug to the development of an adverse event. You cannot quantify the contribution of the suspected drug. Okay, for example, they say that paracetamol induced fixed drug eruptions. Okay, and it is all we are saying in terms of relative terms. Okay, yes, probable. It is probable that the paracetamol has caused the fixed drug eruptions, or it is possible that amoxicillin has caused diarrhea or whatever it is. It is all in a very relative terms. You would not be able to say that yes, amoxicillin has caused the diarrhea, and the contribution of amoxicillin in causing the diarrhea is 70%. The remaining 30% from the other medications or the disease process. You cannot do like this. You cannot quantify that. Okay, that is not possible here. Then final other limitation is it cannot change the uncertainty to certainty. I said earlier advantage, it can classify the uncertainty. It can just classify the uncertainty, but it will not change the uncertainty into certainty. Okay, even after the causality category, the uncertainty remains to be uncertain only, because we do not know what exactly happened in reality. Okay, it is all depending upon some of the assessment, because of the assessment based on the amount of data which you have, you do and categorize. But in reality, what exactly happened in that patient, whether the real is the actual culprit in causing that particular reactions, no one knows. Okay, so that means it cannot change the uncertainty to certainty. So those are some of the limitations of the causality assessment. So there are certain problems in assessing the causalities. So one of the major problem is, that causality assessment is often highly subjective based upon clinician judgment. That's what I said, if you give a same event with the same amount of information to two clinicians, one would say probable and another may say possible. So that means there is a variation in the assessment. Okay, so that means the clinical judgment of the clinician, the clinical judgment of the clinicians, varies. Okay, depending upon who assesses the causality. Okay, so that means it is highly variable. The second issue is that adverse drug reactions frequently mimic other diseases. Okay, it mimics other diseases. That means there is no definite characteristic features of an event, of the reported event, that this is what the characteristic features if it is a drug induced, and this is what the characteristic features if it is a disease as a part of the disease process. No. For example, if you take an headache, whether it is a drug induced headache or it is because of the disease process, the characteristics of the headache remains to be the same. Okay, so that means it is very, very difficult to attribute to the drug. Okay, now the question is why not? It is because of the disease process itself, because even the disease process also it has the similar characteristics of that reported event. Okay, so that means it is very, very difficult to attribute to the drug. That is another issue with the causality. And the third problem is eliminating the alternative causes of suspected reactions. It is very, very important. As I said that, if you want to attribute the particular reaction to the particular drug, you need to rule out all other possible causes, maybe a drug related or disease related. If you have to rule out all other possible causes, means you have to do 101 tests. Okay, but in practical, or in reality, it is not possible to do all those tests. Okay, it is not possible to do all those tests. So it is unnecessary. It is a time-consuming process, and there is a cost involved. Okay, and no one would prefer to do such kind of assessment. So that means, even though you are trying to rule out all other possible causes, in reality, actually you have not ruled out all the other possible causes. So still it remains there is a possibility of the other possible causes. Then another problem is, we know that many of the symptoms attributed to ADR occur in healthy individuals taking no medications. For example, even taking a headache, okay, even a person who is not taking any medications also can develop a headache. So when that is a scenario, how can you say that the headache is because of medication when another patient has taken just because he has taken a medications? You have two scenarios. There are two individuals, one individual without medications, another individual with medications. Okay, the headache, both may develop an headache. Okay, so that means even a healthy person who is not on any medications also develop an headache. So when that is a situation, how can you say that, in other case, it is because of the drug, just because he has taken some medications? That is very difficult to attribute like this when a healthy individual himself can develop such kind of new symptoms. Okay, so that is another problems that are associated with the causality assessment. So these are some of the problems that are, associated with the causality assessment. Okay, because of these limitations and the problems, we know that there are a lot of limitations, okay, in the causality assessment. Okay, in the existing causality assessment case, there are a lot of limitations. And also the causality assessment, if you are doing it with the existing scales, it is, that it is highly variable, it is highly subjective and highly variable, depending upon which scale you use, okay, and also depending upon the amount of information which you have with that, it is highly variable. Okay, so because of these reasons, there is a limited scientific value in whatever the causality assessment which you are doing in the clinical practice. Okay, even if you attribute a reaction to the certain category, okay, but in all probability it is certain, but in reality we do not know. There is always possibility that the drug has nothing to do with that causing that particular reaction. There is that possibility is also there. I'm talking about case by case. Okay, if you are assessing an individual case, there is always that possibility that in that particular individual, the drug would not have caused that reactions, though you have categorized the causality category into the certain or to the probable categories, whatever it is. So therefore, further analytical and other experimental studies are required, okay, to come out with a scale where it is a foolproof, okay, foolproof, and there is to ensure that the existing the drawbacks, okay, the existing drawbacks in the existing scales can be all ruled out, okay, so that a new scale can be developed and that can be applied, at least to ensure that, okay, the assessment what we make, okay, to establish the likelihood relationship between the suspected drug and the reaction is a scientifically more valid. Okay. So with this, I conclude my talk, and if any questions, I'm happy to take it. Maybe at the end of the session, I'm not sure whether now itself or at the end of the session. Now, over to Dr. Rossi.
Thank you so much, sir. That was a very informative session. Thank you for giving valuable insights on the topic assessment, causality assessment of area, sir. Thank you, sir. Without further ado, we can move on to the next speaker, Dr. Juni Sebastian, Faculty, Department of Pharmacy Practice, JSS College of Pharmacy, JSS Academy of Higher Education and Research. Now I request Dr. Juni Sebastian to deliver the talk. Over to you, ma'am.
Thank you, Dr. Rossi. I take the opportunity to thank the organizers for giving me the opportunity to deliver the talk. The slide is just getting uploaded. We'll just wait for 1 minute. Dr. Rossi, can you just tell me whether I am audible and visible?
Yes, ma'am. You're audible as well as your
Can you see the slides?
Yes, ma'am. It's visible.
Okay. Yeah. Thank you. So we'll move on. In this presentation, the next 20 minutes, I'll be discussing you on general introduction, basic requirements for starting an ADR monitoring center, and how to initiate and sustain the pharmacovigilance program, and what can be done or how can we assess the collected data, and what are the possible funding opportunities to open an ADR monitoring center, and a success story of the ADR monitoring center and hospital. I'm not sure whether I'll be having sufficient time to discuss the success story, but we'll just have a look. So the hospital-based pharmacovigilance program can contribute greatly in generating the safety data. So because without a hospital setup, we cannot generate most of the safety data. So basically, the safety data can be generated via the post-marketing surveillance, which is a holistic and comprehensive approach. So we'll just discuss how exactly we can establish an ADR monitoring center in the hospital. A new center in the hospital can start very quickly. So there is no actual requirement for, there is no regulatory description or no actual requirement for starting an ADR monitoring center. You can just start along with any department. We can start any ADR monitoring center. Just you need to have a person who is taking care of the program and some people to report the adverse declaration. That's it. But to maintain or sustain the program, to make the ADR monitoring center very well established and an effective organization, we need to have the time, a proper vision from the organization, dedicated staff, and the staff should be expertise, and there should be continuity in maintaining the pharmacovigilance activities in the hospital. So let us see what are the basic requirements from our experience. So there is a need of a staff to run the pharmacovigilance program. There is a minimum infrastructural requirements, and there is a continuous funding. Let us see in details. And I have seen in some of the chats of the participants, they were asking what are the minimum criteria or for starting an ADR monitoring center, or what is the qualification for starting an ADR monitoring center. There is no such requirements, but ideally there should be a full-time staff should be available for the ADR monitoring center in the hospital. So initially when we are starting, we can have a part-time member, or later we can make it once we have established an ADR monitoring center, we can go with a full-time employee. So for the employee, the basic qualification can be a PharmD, or it can be an MPharm or MBBS, BDS, whichever the medical degree. But we can even nowadays in the first talk, Dr. Rishi Kumar described that the pharmacovigilance subject is incorporated in the BPharm syllabus. So even a BPharm graduate can also be a part of the ADR monitoring center. But we are giving a higher degree because we want to, the understanding of these people would be a little more higher than a BPharm graduate, because he is learning about pharmacovigilance in a very small perspective. But all the other rest, the PharmD, MPharm, MBBS, BDS, all these medical graduates, they would have already done an internship in the hospital, and they would be knowing how exactly the ADR occurs, or they would be having some experience in seeing an ADR or reporting an adverse drug reaction. So we are always desiring postgraduate qualification, and we always look for an expertise in clinical medicine, pharmacology or toxicology, or it can be epidemiology, or it can be always looking for an experience or a trained person than a fresher, and the person should have a good communication skill, should be committed and dedicated to identify and report adverse drug reaction. So the adequate staff, we said adequate staff, the number of staff can be depending on the size of the center. The size of the center, it is the number of reports a center can generate. A hospital, like we can say, ideally, Dr. Ramesh was describing how much time a person will take to perform the causality assessment. He said if all the data is available, it takes 10 to 20 minutes or maximum 30 minutes to perform the causality assessment. So when you see when you are detecting, reporting, and making the causality assessment, it takes minimum 1 hour, approximately 1 hour per reaction will be taken. So in that case, if you are considering 8 hours working time in a day, so that means 8 reports. So if your hospital or the ADR monitoring center is receiving 8 reports in a day, 1 person is required. If you are getting 16, maybe two persons are required. So depending on the number of adverse drug reactions reported to the ADR monitoring center, we can decide the total number of staff required for the particular ADR monitoring center. And always there is a secretarial assistance can also be needed. So that means a minimum 2 members are required for establishing an ADR monitoring center. So while taking the staff, we have to taking care of the educational training of the person. So he should be properly trained on data collection and verification, interpreting and coding of adverse drug reaction. So generally the WHO ART or ATC, so different coding sections, coding is available for adverse drug reaction and the diseases. So somebody who is knowing little about the interpreting the coding of adverse drug reaction has to be there in the ADR monitoring center. The coding of drugs, he should be knowing how to do the causality assessment, how to detect the signals, and how can we assess the risk management. So this person has to be either educated or trained in these different areas of pharmacovigilance. So coming to the infrastructure, the location can be at any department, can be associated with any department in the hospital. It is not necessary that there should be a separate pharmacovigilance department. And I don't think most of the ADR monitoring center are attached with any of the different departments in the hospital. The ideal departments to work in collaboration with pharmacovigilance is, it can be clinical pharmacy, it can be clinical pharmacology, it can be clinical toxicology, or it can be epidemiology departments. So it's always preferable to have to attach with the department which is working in the hospital, so that the staff of the department who is already there, other than the pharmacovigilance staff, the other staff who works in the department is already there in the hospital setting, they can contribute little to this pharmacovigilance center. And it is always preferable, it can be located in the middle of the hospital somewhere where all the healthcare professionals can access it. Or it is not necessary that it should be in the any accessible area. If the center has phone facility or internet facility, it can be anywhere. There should be adequate space. There is no structural requirement, or there should not be this much spatial area is required, nothing like that. There should be, the space is adequate to house the staff and the infrastructural requirements. So the other requirements for infrastructure requirements are, there is a communication systems are required, like the computer, internet, telephone, fax, printer, and photocopying machine. And there should be a library or information system. And to understand, okay, because though you are expecting, you need to understand whether this drug will cause some kind of ADR, what is the mechanism of this adverse drug reaction, or how long it will cause. Because generally when a doctor is reporting an adverse drug reaction, if it is a very recent one or he has not experienced it before, he will definitely ask you, how can we treat this situation? Or if I stop this drug, which other alternate drug can be given? So to give these kind of information, the pharmacovigilance center need to have some information system. So he can have standard books, or it can be some literature resources which is related to adverse drug reaction. Or we can have, other than the standard textbooks, we can have some journals or indexing services we can have. Or if that is not available, if it is a teaching hospital, if the area pharmacovigilance center is associated with a teaching hospital, then we can directly use the hospital library, which would be already having all these kind of facilities. Then there is small office equipments are required, like a computer chair, table, then office chair, cupboards, bookshelves, journal racks, all that. And that particular area has to be adequately ventilated and for the proper function. Then once we have all these facilities, we have a dedicated staff, we have attached with a good department, we have a small office, which the requirements. Then how can we initiate the process? Because we want to make the healthcare professional know that there is an ADR monitoring center functioning in the hospital. So how can we do that? We can do that through the awareness seminars, we can conduct workshops, we can have training meets for the healthcare professionals on the importance of adverse drug reaction. Because initially, many of them may not be knowing what is the need for reporting adverse drug reaction, what all events can be reported, how can we report, whether it is through a reporting form or we can use an electronic reporting. They won't be knowing all that. So we can conduct some kind of workshops and training programs for all the healthcare professionals. So when we are conducting, we should be conducting in section-wise. Maybe the information which can be given to doctors may be different to the nurses. We need to give a little more detail. So section-wise, we can conduct these pharmacovigilance training programs and in a phased manner. It is not at one time, because in the hospital, always the staff will be some of them will be leaving and new staff will be joining. So it is a continuous process. We have to keep conducting these webinars, workshops until they are knowing that there is a pharmacovigilance center is present in the hospital. And we can design an ADR reporting form for the collection of data. So in the previous speakers, they spoke on different ADR reporting forms issued by CDSU under the Pharmacovigilance Program of India. There are 2 reporting forms that are available. One is for the healthcare professional and for the consumers. So if you are feeling you can directly go ahead with the ADR reporting forms, then you can go ahead with that. Or you can make a minimum we can condense that minimum information and you can collect the data. In our hospital, what we have done, we are not using directly the form which is given by the Pharmacovigilance Program of India. Instead, we have created our own reporting form. So our reporting form has very minimum information: which drug has caused the ADR, what time it has caused, and what event has caused, and which doctor is reporting, and the patient's IP number. And we have placed the ADR drop box in the different units in the hospital. So they will fill these minimum information. These forms are made available in all the nursing stations, and all the doctors know that these forms are available in the nursing stations. So what they do, they will directly fill the form and they will drop in the box. So the pharmacovigilance associate who is working in the hospital will be opening these drop boxes every day and will try to identify the form ADR reports. Then from this IP number, he will track the patient and he will find out the information. So likewise, we have improved the reporting rate. Otherwise, the doctors may feel, or the reporters may feel that there is so much to fill in the form, so they don't have time and they don't report the ADR. And we have to keep the reporting form in an easy access, and we have to provide a feedback what has happened to the report, whether we have reported to the ADR AMC. When the previous speaker, Dr. Pramod, when he was narrating, he said there is a feedback generally we will receive from the PVPI. So when we are receiving a PVPI in the ADR monitoring center, we can give that information to the reporting doctor saying that we have reported the event. So, and we can initially when they are reporting the ADR, we can give a feedback and can also give a thank you notes. And we can establish a database for the easy documentation and retrieval of information. It's very easy. We can our own database we can create. It may be an Excel sheet where we can note all the information. So it will be helping us to understand what kind of ADR is reported. Otherwise, what happens is when the hospital is having an AMC center, we will get access to Vigiflow, which is the online platform where we are reporting the ADR to the PVPI and from there to the International Drug Monitoring Program at Uppsala Monitoring Center. So once we have reported, the soft copy will be directly going to the Uppsala Monitoring Center. So we can have one more database for our understanding and review. And we can have some kind of contact with the national and international institutions working in pharmacovigilance so that they will help us or they will support us in conducting these kind of training programs. It can be our PVPI, it can be the WHO Department of Essential Drugs and Medicines, or even the Uppsala Monitoring Center. Now we initiated the process. To sustain this, we have to keep conducting the campaign programs, we have to disseminate different materials, and we have to give the information on what happened to the ADR report and which is it recently understood ADR from our center. Likewise, so we can keep continuing the education programs for the healthcare professionals to sustain this program. So best, though there are various reporting methods available, the best method is spontaneous reporting form. So when you are saying spontaneous reporting, any healthcare professional, whenever they identify an ADR, they can directly report to the ADR monitoring center. So the best method which can practice in the hospital is a spontaneous reporting form, and we can give the reporting forms, and even through electronic reporting, they can report it, or they can send a message, or they can send an email, or they can fill the ADR reporting form. So make all these provisions open for the ADR monitoring center. So we already discussed this. Any healthcare professional, even the consumers, can report the adverse drug reaction. So it is not necessary that there should be a eligibility criteria to report an adverse drug reaction. Anyone who works in the hospital or the patient, the consumers, can report the adverse drug reaction. and any suspected adverse drug reaction can be reported. It is not that like a chemotherapy-induced nausea, vomiting. It is a non-reaction. It can be unknown reaction, serious reaction, mild, moderate, any kind of adverse drug reaction can be reported to the PVPI program. When you are reporting this adverse drug reaction, we can change the frequency of ADR. And most of the time what we observe is under-reporting of adverse drug reaction. Most commonly, it may be a technical or a psychological issue. So doctors or the reporters may feel that when I report the adverse drug reaction, others may feel that it is my fault, or they perceive that it is my fault that the drug has caused the adverse drug reaction. So that is a common problem that lead to an under-reporting. And there is a fear of negative competence, there is a possibility of litigation, or there is a lack of knowledge what happens after reporting, or they won't be really understanding what is the system of reporting the adverse drug reaction, all that. And there is underestimation of true size of the problem is also another commonly seen problem among the healthcare professionals. So these under-reporting, how can we stimulate? We can keep the ADR reporting, we can condense the ADR reporting form, we can have a small ADR reporting form, we can make it available at very easy access places, and acknowledge the report of adverse drug reaction. Even when we are initially starting, we can make phone call, because if we are having only 2 or 3 reports, it is very easy for us to call and acknowledge to the doctor or the reporter that we have received the report. Or we can provide feedback, or if it is a new reaction, we can, keeping him as an author, we can publish the case reports or case series, or we can publish it through the newsletters of the hospital. And participation of center, like the ADR monitoring center staff, like UG students, PG students, we can involve them. And in their meetings, we can go and stimulate them. We can tell them what is happening. And we can collaborate with local and national pharmacovigilance centers and professional associations. And we can have an ADR assessment committee. So this committee will help us to assess the data, interpret the data, and make new publications. And when you are assessing the data, we have to look for different things. The quality of documentation, whether all the fields, which is a mandatory field, is, or all the details are filled. If all the information is available, only then we can do the causality assessment. So we need to check for the quality of documentation and whether the drugs are properly coded as per WHO ATC, anatomic therapeutic chemical classification of drugs, whether the adverse drug reactions are coded as per adverse reaction ADR, WHO ART or MIDRA. Then we have to identify is there a duplication of reports. That is most important. There maybe we may receive 2 reports, one from the nursing staff, one from the pharmacist. Both might be of same patient. So you can identify that from the IP number of the patient. So those kind of, we have to identify the duplicate reports, and we have to perform the causality assessment. And the reporter, there is no need for the reporter to know the ATC and ART. He can just give the drug name and the adverse drug reaction. It is a duty of the technical assistant, the staff who works in the pharmacovigilance center, to code these the drug for using the WHO ATC and the WHO ART. And these data, once we have reported from the ADR monitoring centers in the current scenario, from there it goes to the national center, PVPI. There, these data will be used for hypothesis generation or strengthening already generated hypothesis. And as we discussed already, drug regulation, maybe withdrawing the drug from the market, or making some black box warnings, etc. Drug regulation and dissemination of information. Like we will get some information, that information can be passed on to the healthcare professionals so that we can improve our practice. Then again for the education and feedback. And the ADR monitoring center can have a good relation with the drug regulatory authorities, pharma companies, then the different professional associations, national centers, international centers, then academic associations or academic hospitals or the nearby pharmacy colleges or whoever it is, and the media and consumer organization. And lastly, the funding is the most important thing. And we have to understand when there is a dedicated staff and a secretarial staff, and there is a space and a computer, all that, to function all these, there is a fund generally required. So without the funding, we cannot operate this pharmacovigilance program. So there are recurring cost, non-recurring cost, and maintenance cost. So we have to calculate or estimate the cost before we start the program. So we can look for funding, maybe from the university departments, maybe it is associated, the hospital is attached to the university, then we can look for funding from the university, professional association, even from the government, NGOs, non-governmental organization, or some health insurance companies. Some of these we can look for the different kind of agencies. Likewise, we can get some kind of funding. Or the cost can be minimized if the PV program is incorporated to some other program. So like the Pharmacovigilance Center is working in association with a Drug and Poison Information Service, then we can improve. That is what we have done in our hospital. The Pharmacovigilance Center is working actually with the Poison Information Center.
Dr. Judy, ma'am,
So, yes.
Ma'am, like within 1 minute, can you wind up the session?
Yeah. I'm done. Okay. So we have initially we had. Yeah. Understand. I understand. So initially we had funding for the Pharmacovigilance Center, but later we did not have funding for Pharmacovigilance Center, but we got a funding for Poison Information Center. So likewise, we collaborated both and we started functioning. So this is what is the thing which I was looking for. I wanted to tell you, this is the ADR Monitoring Center. It is working in the JSS Hospital. This is the Department of Clinical Pharmacy. We are working along with that. So just want to tell, it is managed by Clinical Pharmacy, which was already described by Dr. Pramod Kumar. We started this program in 1997, and we are working with JSS Hospital. Like the program is introduced in 1997, and we are working with a 1,800 bed teaching hospital. And we are currently the regional training center for South Zone. We have collaboration with Uppsala Monitoring Center for
Dr. Joe, Rosie can request Dr. Priyank to continue.
Yes, okay. Now I request Dr. Priyank Tripathi, Clinical Pharmacologist and Manager, Healthcare Global Enterprise, Bengaluru, to deliver the talk. Over to you, sir.
Thank you so much. Thanks a lot. Just give me a minute.
Sir, within 15 minutes, please.
I will. Yeah. I'm keeping a tab.
Okay, sir. Thank you.
A very good afternoon, everyone. Thank you so much. Thanks a lot to one and all for inviting me, and it's indeed a privilege to be part of this program. Looking into time, I hope I'm audible and my presentation is visible. So my sincere thanks to everyone. Sincere thanks to JSS. Sincere thanks to Dr. Joshi for remembering me and inviting me here, and my thanks to Kidwai Member Institute of Oncology. My thanks to inviting me out here. Am I audible? If somebody can give me the feedback.
Yes, sir. You are audible, sir.
Thank you so much. So looking into the oncology perspective, I think you all would have been primed with how do we track ADRs, or what pharmacovigilance is, and all related aspects of establishing the pharmacovigilance center, from assessing a causality of event. So let us look into how and why the pharmacovigilance is important in oncology. So I'll quickly run through. I'll try to quickly run through my presentation. So before I start, the views I represent here solely belongs to me and does not represent anything from my parent institution, which is HCG. Well, why in oncology? So there are various reasons, but if I can enumerate few, these are few. The drugs which we use belongs to narrow therapeutic index. Yeah? And the drugs which we give, we try to give in maximal tolerated dosages. We do work upon the PK/PD concepts in these drugs and try to use the maximal possible dosages for those patients to get the optimal therapeutic benefit out of it. Now, when we do so, we left with very minimal very minimal concentrations or very minimalistic kind of concentrations to play around, and the kind of adverse effects or the kind of toxicities which might come up, sometimes may be grave. So that's what the next point talks about, the basic nature of these drugs. These are cytotoxics, most of these drugs. Either we use biological agents or we use the core cytotoxic agents. The primary aim of these drugs is cytotoxicity. Primary aim of these drugs to initiate the down regulating mechanisms so that we do not allow the cancer cell to grow further. So that's the basic nature. Apart from these drugs going and apart from those who target certain targets, these drugs in general go and hit basically all the cells wherever they can reach. So that way, they have this intrinsic biologic toxicity associated with them. So even if we use them and we know that we are using it in the maximum tolerated dosages, so it becomes really important to monitor our patients. It becomes really important to systemically see what is happening in our patient in terms of safety, in terms of the adverse effects which these drugs may precipitate in a patient. Now, another aspect which makes the whole concept really important in oncology is about the timing of treatment regimen and the combination regimens which we usually form. Well, we all understand, most of us understand that whenever we are treating a patient of cancer, we give the drugs in certain sequences. We either give it weekly or 2 weekly or 3 weekly or 4 weekly or every 30 days once and so on. And when we do so, we give it those drugs in combination. Well, it may happen that patient might have to skip one or the other dosages or cycles in between. Now the question comes, when to introduce the next cycle, and if we are introducing the next cycle, whether will it affect with the toxicity profile of the drug, or if it is a combination regimen, should we withhold the whole cycle, or should we withhold a particular part of or particular drugs from that particular regimen? So if we do so, what will happen if I introduce or if I take a drug out? Will it impact my patient in a negative way, apart from helping him therapeutically? So this particular aspect really makes it complex and makes it important for us to see that we monitor our patient regularly. We take feedbacks from them if they are facing any drug related problems in terms of the adverse effects, and give a feedback to the treating teams that your patient A, B, or C has come up with so and so adverse effect, or has communicated so and so with us. Well, the drugs do have an accelerated adverse effect profile. Moment we go beyond a particular dose, we see that the patient starts precipitating one or the other adverse effect, and sometimes within the therapeutic dose itself, they start precipitating certain adverse effects, which again makes the whole treatment modality, the whole treatment paradigm little complex. And again, it is important that we take a note of every single event. It might be minor, but we take a note of every single event. Well, it is important because each and every treatment protocol is individualized. Yeah, each and every treatment protocol is for one particular individual. There might be 2 patients with the same diagnosis for the same protocol, but their dosages might change. Sometimes for those patients, depending upon how well they are able to tolerate the treatment, their frequency of treatment, their timing of their drugs or the timing of their drug cycles may change. Well, this is all because of this accelerated adverse effect profile of these drugs. Well, the next point is of real importance. This is something which we evolve, very frequently, especially in oncology practice. We try to add on new therapies based on based on the trial experiences, based on guideline recommendations, and based on our own experiences on the patient and understanding of the drug therapeutics. Well, the most common thing out of that is adding biologicals along with the cytotoxic agent. This might be incorporated as part of the drug, or this can be introduced separately as 2 individual agents. Now, when we do so, when we are introducing a biological with a cytotoxic, we are basically introducing an adverse effect of a biological plus an adverse effect of a cytotoxic along with the therapeutic effect. Well, at times, if there is something which is common with both the agents, we basically may have to monitor our patient really closely, and may have to vouch or may have to watch, in fact, for any of those adverse effects which might affect our patient and which might be detrimental in deciding the fate of our therapeutic plan, basically, and also the failure or success of our therapeutic strategy. Well, another very important and dynamic aspect are the new indications which get approved, very frequently for a lot of therapeutic agents. So when I say so, currently we see a lot of tyrosine kinase inhibitors in oncology practice, which are getting, which are being approved for new indications within oncology. We do see a lot of, immunotherapy agents, the biological immunotherapy agents, the checkpoint inhibitors, wherein we initially started with myeloma and renal cell carcinomas, and today we have, of course, the lung cancers. And today we have plethora of indications being approved for the checkpoint inhibitors or the immunotherapy. Now, when that happens, large population usually get exposed to these agents, and then we come across, it becomes really important that we monitor all these patients, because the basic nature, as we saw above, the basic nature of these drugs is toxicity. So we have to monitor our patient and see, whether which subset or which subgroup of patients are really experiencing it more, or which subgroup of patients are not experiencing minor adverse effects, or is there anything in particular which can be done to minimize it? Yeah, and it is important that we report it. Until unless it is not reported, until unless it is not shared and discussed, will not come to know about the incidence of these events and the severity of these events associated with these drugs. Well, a lot of these cytotoxic agents, less of biologicals but more of cytotoxic agents, do have a lot of potential of drug interactions, because these drugs, in particular the cytotoxic, the tyrosine kinase inhibitors in particular, if I talk, do get metabolized through CYP enzyme family, and I hope we all understand the importance of this enzyme family in the pharmacokinetic profiling of a drug. Well, I'll not go in depth on this on that aspect. The last point, the metabolic pathways which these drugs follow for their metabolism primarily. So, for example, I'll talk about one very famous and a very old drug, 5-fluorouracil. Now, 5-fluorouracil, gets metabolized and changed into its cytotoxic form through DYPD. Well, the individuals who are deprived of this particular enzyme, may have higher incidence of 5-fluorouracil toxicity. Now, it becomes again important for us to see whether any of our patients are experiencing, accelerated toxicities with 5-fluorouracil. If yes, then it becomes again, it really becomes important for us to see whether do we have to check our patients for DYPD. Well, for the students who are part of this program, this is something which we come across every single day, the adverse effects which are associated with these cytotoxic agents, right from cardiovascular effects to the immune-mediated reactions or the adverse effect profiling, to the venous adverse effects with that of thrombophlebitis, extravasation, etc., cognitive dysfunctions at times which we see, we name them as chemo brain or chemo fog, the mucositis, infusion-related reactions, thrombosis, which again is something which is seen in cancer patients, and does require monitoring. All these patients does require monitoring for thrombosis, does require monitoring for any of these cognitive dysfunctioning, for neuropathies, nephropathies, for endocrine toxicities, etc., and cardiovascular toxicities, etc. Well, I was talking about the accelerated approvals. If we look at for the matter of fact, oncology is one of the rapidly growing domain in terms of drug discovery. So there are a lot of accelerated approvals which are being given to many oncology or anti-cancer drugs primarily. If we look at the numbers in 2019 for the whole year, we had around 10 drugs which got accelerated approvals. So when we say accelerated approvals, you all understand that the drugs been approved, before the time frame when it was expected. Now, which means that we probably would have missed certain safety profiles of these drugs while we were testing on the patients, or while the drug was in the active phase 3 trial. It becomes extremely important that we monitor all these drugs throughout the time whenever we are using it, in case if we come across any such events. I was talking about new indications being approved very recently, mitomycin being approved for a new indication, tyrosine kinase inhibitors now and then are being approved for various new indications depending upon their their safety. And again, it becomes important for us, especially for both these drugs, the tyrosine kinase inhibitors and the immunotherapy, the checkpoint inhibitors, that we monitor our patients for various immune-related adverse effects. Apart from the causality assessment scales, which are being explained to you in the previous sessions, the criteria which we primarily use for evaluating the adverse effects here is the CTCAE criteria, which we call, which is common terminology criteria for adverse events, wherein we grade these events from grade 1 to grade 5, grade 1 being the mild and grade 5 being death. And how does this program helps? Now, I'll give few examples from some of the very rare events which we reported in our own system by our team. 3 of those significant events, 4 of those, in fact, we did identify patients, experiencing hyperglycemia when they are on lenalidomide therapy and sorafenib therapy, and hypomagnesemia when the patients are on bevacizumab therapy. If you see the incidence in the literature, you might not find a very high incidence of these events, but it does require close monitoring of your patient to identify it and communicate it to the primary treating team so that necessary steps can be taken in future to mitigate these. Next are the drug-induced Ogilvy syndrome with cisplatin and oxaliplatin-induced sinusoidal obstructions, which the team was able to identify, report, and then we are able to strategize the monitoring strategies for our patients. So how does it help is, it really helps in modifying or introducing new supportive care therapies and the observation strategies so that we make our therapeutic plans more safer, we make our therapeutic plans more useful for our patients primarily. Yeah, more safe, more useful is what we want. So thank you so much. Thanks a lot for inviting me here. That's it from my end. If there are any questions, I'll be more than happy to take, or else you can write to me at my email ID, which is written over here. Thank you so much.
Thank you so much, Dr. Priyank Tripathi, sir, for covering up within small minutes, like within small duration, you have covered up a lot of information. Thank you so much, sir. Thank you for sparing your valuable time with us. Sir. Thank you. Now I request Dr. Spruha Dalakia, Assistant Professor, Department of Transfusion Medicine and Blood Bank, Kidwai Memorial Institute of Oncology, Bengaluru, to propose the vote of thanks. Over to you, ma'am.
Thank you, Dr. Rosie. Am I audible?
Yes, ma'am. You're audible.
So it is a very happy moment. Kidwai Memorial Institute of Oncology, Bengaluru has organized this extremely meaningful and need-of-the-hour workshop on pharmacovigilance in association with Sarada Vilas College of Pharmacy, Mysuru, Karnataka Government Medical Education Department Institution, Employee Welfare Association, KMIO Unit, Regional Training Center for South Zone Pharmacovigilance Program of India, Department of Clinical Pharmacy, JSS College of Pharmacy, JSS Academy of Higher Education and Research, Mysuru. I thank the Chief Guest, Dr. C. Ramchandra, Director, Kidwai Memorial Institute of Oncology, Guest of Honor, Dr. T.M. Pramod Kumar, Principal, JSS College of Pharmacy, JSS Academy of Higher Education and Research, Mysuru, Convener, Dr. Hanumanthachar Joshi, Sarad Vilas College of Pharmacy, Mysuru. I also thank Dr. Dattatri A.N., HOD and Professor, Department, Karnataka Institute of Medical Science, Hubli, for the welcome address. I have no words to thank the faculty of presenters, which include Dr. Rishi Kumar, Dr. A. Pramod Kumar, Dr. M. Ramesh, Dr. Harsha Chalasani, Dr. Juni Sebastian, Dr. Priyank Tripathi, and the respective organizations. I extend sincere thanks to IT team of all these institutions. This workshop is successful due to the participation of delegates. They deserve a great round of applause. Dr. Rosie Anand requires a thundering applause for excellent steering of the program. A great thanks to many more who could not be named here because of the want of time. We are hopeful we can conduct some